Good morning, everyone. My name is Lian Wozuel. I am a researcher and guest lecturer at Latvia University of Life Sciences and Technologies in Faculty of Food Technology, and it is my honor today to be your chairperson for this section in this 28th Annual International Scientific Conference, Research for Rural Development. It's my honor to be here today, also as a speaker, and introduce you and your researchers. I guess before we start, just a quick reminder for the speakers. From technical point, you have 10 minutes for your presentations and then 5 additional minutes for discussion we will have. Please make sure that you, when giving your presentations, share your entire screen, not just the presentation, so we're able to see, so everyone can see your presentations. And also switch on your microphones when you are speaking, and please switch them off when you are not. So I guess if everyone's ready, then maybe we could start today with our first participant. I would like to introduce Daiga Konrad and her research on extraction of bioactives from pumpkin byproducts and determination of their antioxidant activity. So I can see Daiga's presentation, so I guess she is ready.
Nice to meet you on the 28th already conference for research for rural development. And I'm very happy to introduce you with part of my postdoc research about use of byproducts for extraction of bioactive compounds. And I am Daiga Konrad from Riga Technical University Institute of Organic Chemistry, and my colleague Professor Kris Spalviņš was involved very much with equipment and knowledge in this research. And so let's go on with the research. And as we know, during food production, as well as in agro-industry, there are huge amount of waste. And we are not only talking about waste in household, but in primary production too, such waste as peel, pomace, seeds, and etc. In Latvia, most popular vegetables and fruits are apples, plums, berries, and as well as pumpkins too. Pumpkins are used in juices, purees, jams, and therefore byproducts can occur in every step of food production. And byproducts already can be up to 70, almost 90%. And byproducts are peel, bark, pomace, seeds. And for example, in some regions of Europe where pumpkins are used for oil production, like Styria, Slovenia, and Turkey, there are used special varieties of pumpkins, and therefore for oil production, they use only seeds, and therefore pomace and peel are as leftovers and can consist of 90% as food waste. And nowadays there is an increasing interest in natural antioxidants such as carotenes, saponins, tocopherols, fatty acids, and pumpkins are very rich in these bioactive compounds, and also their anti-radical activity is very high. And for extraction of these bioactives, there should be paid attention for solvents we use for extraction, and according to these new guidelines in green chemistry, we should pay attention also to solvents we use, and therefore subcritical carbon dioxide extraction technology can be widely used as an alternative for conventional extractions. And the aim of or the goal of the research was to use pumpkin peel and seeds cooled with all, not only the seed, this inside, for extraction of bioactive compounds and to determine antioxidants content, profile, and anti-radical scavenging activity of these extracts. For extraction or for experiments, we use two varieties of pumpkin, pink banana jumbo from Lot Eco Food Rudolph's, which is a producer of baby food. Therefore, the waste product was seeds and peel. All of them are eco-certified. And for the second or other variety was Muscade de Provence from farmers Kliblapsas in Kurzeme. We used these byproducts, and there we can see methods, how did we prepare them for extractions, and all materials were prepared in the Institute of Horticulture in Latvia. With these equipment they gave us to prepare these materials. And these extracted materials were analyzed for a total content of carotenes, beta-carotene, lycopene, lutein, zeaxanthin, and for pigments, chlorophyll A and chlorophyll B. And also for anti-radical scavenging activity, we used the PPH assay and analyzed with UV visible spectrophotometer. Different solvents as well were used for analyzing, and there is the schematic attachment of extraction and determination of carotenes and pigments and anti-radical activity. For extraction, we used subcritical carbon dioxide extraction with Fanax extractor. There you can see the conditions we used for extractions. We used Soxhlet extraction, and the solvents were used ethanol and hexane. The temperatures for extractions were from 60 to 70 degrees of Celsius. And we used also for carotene extraction edible vegetable oils as rapeseed oil, coconut oil, olive oil, and grapeseed oil. And for oil recovery, we also used oil press. It's a single screw extruder type oil press. The temperatures for extraction also were rather high. And for determination of biologically active compounds, as carotenes and pigments, we used different solvents, acetone-hexane mixture, acetone, cyclohexane, and ethanol for and we used different absorption wavelengths. And for anti-radical activity, we used diphenyl picrohydrazine radical solution. And there are results we obtained in our research. The oil content differed from varieties. The highest content was in Muscat de Provence. This is the variety we used from Lateco food, as from Lateco food byproducts. And as we can see, the highest yield was received with Soxhlet extraction using hexane as solvent, and it follows with subcritical carbon dioxide extraction. For experiment, we also analyzed carotenoids in edible oils, which we used as solvents. There we can see in table number 1 the content of total carotenes, lycopene, beta-carotene, lutein, and zeaxanthin. And as it's seen that stability of carotenes is very sensitive, and during the sunlight and temperature exposure, they can form isomers, and then that's rather hard to determine these exactly some carotenes. And rather different results were obtained with cyclohexane and acetone as solvents. And acetone as solvent for detection of chlorophyll showed better results in comparison with hexane acetone. And also we determined chlorophylls A and B with acetone and acetone hexane mixture. Therefore, it was necessary to find what solvent can be used for detection of pigments. And some solvents gave negative data in determination of chlorophyll. And lutein and lycopene. There you can see sample abbreviations where we can see how we extracted these oils.
I'm sorry, I must apologize, but your time is up. Could you please maybe quickly summarize your research?
Okay.
So,
yes,
yeah,
thank you.
Thank you.
Yes, and for composition, we also analyzed, and there was no significant difference in determination in chlorophylls with acetone and acetone hexane. There we can see assay of chlorophylls, carotenes in extracts of pumpkin byproducts, and carotenes in edible oil extracts. And anti-radical activity was the highest with coconut oil extractions. And for conclusions, we conclude that high-value bioactive compounds have been extracted, and the subcritical extraction is effective green solvent, as well as vegetable oils are effective green solvents. And there is acknowledgement for our postdoc European Regional Development Fund. And thank you for your attention. And any questions, please?
Okay, maybe just a quick question regarding your research. Thank you very much. It was very interesting. what possibilities do you see for these extracts to be used in everyday, maybe use new product development or something? Is there a perspective to that maybe?
Yes, thank you very much for this question. I waited for this question because these extracts for the next stage of research will be used for food industry, for fortification of edible oils, and for maybe for cosmetic products in using them as anti-aging creams or sun protective creams, while carotenoids are very good scavengers of this oxygen.
Okay, thank you very much for that. Does anyone else have any questions?
Yes, I have also a question to Daiga. Hello, everybody. Hello, Daiga. I'm happy that you continue your research activities, and your research activities are really very close to your PhD thesis. My congratulations. But my question is, do you analyze what kind of solvents are allowed in food industry? Because you have a lot of solvents and some of them are prohibited. Did you take into account it?
Yes, I did it. Thank you, Inga, for your question. Yes, there are very small amounts of organic solvents that can be used in food industry or also in cosmetics. That's why I'm trying to find these solvents, as we call them, green solvents that are allowed, not prohibited in food industry.
Mm-hmm.
Yes,
Yeah,
but
yes,
I used only for determination of carotenes and pigments, these prohibited solvents. That's why
No, I'm just pay attention because we know that the chemists like very much hexane and then some similar.
Yeah. That's why I'm searching for green solvents as subcritical.
Be very careful about these hexanes because we have some specific rules when and then how we can use especially.
That's why I'm trying to extract.
For the acetone is little bit more application and concentration as well. But okay, my congratulations, and I'm happy that you are here. And good luck, and always.
Thank you, Inga.
If there are no more questions, then I would like to move on to our second speaker, which is Alina Volusco. Are you here, Alina?
Hey, yes, I'm here.
Yes, and her research review on the recent advances in the analysis of human milk oligosaccharides. Please, the floor is yours.
I would like to talk about oligosaccharides in human milk and latest achievement in human milk oligosaccharides analysis. So, as you can see in breastfeeding statistic graphic in Latvia, the trend of breastfeeding is increasing from 2010. Breast milk is a unique and ideal source of nutrients for most infants. It contains a lot of bioactive compounds such as protein, vitamin, nucleotides, minerals, oligosaccharides, and immunoglobulins. Human milk is classified as the first functional food in the infant life. The composition of breast milk in Latvia has been studied fragmentary up to 2016. However, there are currently no studies about oligosaccharides in breast milk in Latvia. So the aim of the study is to find out recent advances in the analysis of human milk oligosaccharides, and tasks is to analyze the latest information about the recent advances in the analysis of human milk by liquid fast separation methods, to investigate any known association between human milk oligosaccharides composition and maternal nutrition, to investigate the effect of human milk oligosaccharides on the infant's development and health. The database of scientific paper, Web of Science and Scopus were examined in order to find out data from the latest achievements related to analysis of breast milk oligosaccharides, which were published from 2011 to 2021. The search was based on two key terms, human milk oligosaccharides and human milk oligosaccharides analysis. Human milk oligosaccharides have effect on the infant development and health. It recognized as one of the most important bioactive factors that change the outgrowth of genes in immune cell as immune modulator in the gastrointestinal tract work as probiotic. Such a search of nutrients as human milk in infancy, in which human milk oligosaccharides are present among different bioactive compounds of human milk, is recognized as dangerous for healthy microbial colonization of infant. Most with immune component diseases and infection, including diarrhea, urinary and respiratory tract infection, bacteremia, and others, occur less often in infants than in children who were fed by infant formula. Few scientists have investigated the effect of maternal diet on the bioactive compounds of human milk oligosaccharides. Client and other work focused only on dietary components, but as a result, it turned out that there was a relationship between diet, total sugar dietary fibers, and focused galactose in human milk oligosaccharides. Seferovich and others found that changes in maternal nutrition replenished of carbohydrate sources lead to change in some major component of human milk oligosaccharides. Christine and others found in their work that it turned out the milk microbiome is changing if you follow a diet high in fat and carbohydrates. High fat content reduces the concentration of sialated human milk oligosaccharides and compared to galactose, glucose significantly alters the concentration of fucosylate human milk oligosaccharides. In latest information about the recent advances in the analysis of human milk oligosaccharides by liquid phase separation methods, I found that human milk research began in the early 1900s. Before starting the analysis, human milk oligosaccharides need to be isolated from other components that are found in human milk, such as lipids, protein, and lactose. Currently, method for preparing human milk oligosaccharides samples comprise similar steps, typically starting with removal of lipids and protein. The degreasing step is often carried out by centrifugation or solvent extraction. Protein precipitates tend to complement organic solvents such as ethanol, chloroform, methanol, acetone, or acetonitrile. The proposed method for analyzing human milk oligosaccharides are larger based on liquid chromatography. At the moment, a large number of high-performance liquid chromatography methods have been identified which are used for the analysis of human milk oligosaccharides. Pluses of this method is that high performance liquid chromatography can be used with label-free detection modes. The method of obtaining a sample for the analysis require only dilution, ultrafiltration, or centrifugation. And the greatest drawback of this method that are based on liquid chromatography is the need to use oligosaccharide standards, and standards are often very expensive. For human milk oligosaccharides analysis by capillary electrophoresis, pluses is that strong glycoanalytic tool due to low sample volume requirements, reduce buffer use, height, and fast. Electrophoresis provide several separation modes that are easy to change during operation. And minuses is that in working with capillary electrophoresis is needed primary for unchanged carbohydrates in order to provide them with the necessary electromigration. One more method to analyze human milk oligosaccharides is mass spectrometry. The pluses of this method is that during the mass spectrometry ionization period, the sample is mixed or covered with an activity absorbing matrix to separate the carbohydrates, and then ionize with a laser. So the conclusion is that human milk oligosaccharides helps to develop many beneficial properties in the infant body. Human milk has a beneficial effect on breastfeeding. The analysis of human milk oligosaccharides is considered very complex, that heterogeneity and different isomeric, anomeric structure. And the proposed method for analysis human milk oligosaccharides are largely based on liquid chromatography. That's all. Thank you for your attention. Maybe you have some questions.
Thank you for your presentation. It was very nice. Questions, anyone? No questions? Maybe then I will ask one. So very interesting subject. What are your own personal kind of aim for research further on? You've done the review. What are you planning to do next? Are you working with the same subject?
Mm-hmm. So the topic of my dissertation is bioactive compounds in human milk and its association with maternal diet. We study minerals and oligosaccharides in breast milk. Mineral analyses are performed in the bio laboratory, but we plan to analyze oligosaccharides in our university by chromatographic method. This review is very important for me to start the analysis of oligosaccharides. For example, now we attracting research participants, obtaining milk samples for study from participants. And in these moments, we have 30 samples of milk to analyze human milk oligosaccharides. That's why this review is very important for me. It's like a first step in analyze human milk oligosaccharide.
Okay, thank you. If there are no more questions, then maybe I will give the word to Iveta Rutka. Iveta, are you ready?
Hello, everybody. Bee drone larvae are one of the byproducts of beekeeping that need to be monitored for chemical composition and microbiological contamination. Bee drone brood homogenate is produced from drone larvae as a byproduct of beekeeping. Bee drones eat a significant amount of honey from the hive and gives losses of honey to beekeepers. It's difficult to determine the age, actual age of larvae in beehive during harvesting. The 10th day, the bee drone cell is captured and can be used to accurately determine age. There we can see captured and uncaptured bee drone cells. Bee drone cells are much larger than working bee cells. Warm mite is a global problem for beekeepers around the world, as it can lead to the death of a bee colony. The development cycle of bee drone brood is longer than the development of working bees. Therefore, warm mites is developed in drone brood cells. To reduce the production of the warm mite, drone larval removal is usually done in the spring, often in April, May, and June. During the collection of bee drone larvae, the presence of warm mite was detected in the captured drone cells. Warm mite attached to the body of the larva and weakness it by sucking fat bodies with gut husoma. Microbiological contamination of the harvested larvae is caused by individual motley larvae. The warm mite crawls on the larva in the cell before it's captured. If one larvae is infested with several mites, he dies and begins to mold. The aim of the work is to harvest bee drone larval samples by different techniques and to determine the chemical composition and microbiological contamination. Two different bee drones larval harvesting methods were developed in the study. The first method was to take the larvae with tweezers from the comb of the bee cells right next to the hive. At the time of removal, the larvae immediately died and began to deteriorate. This was evidenced by a change in color tone. The larvae were then compressed and homogenized. The homogenized mass was placed in plastic container and then frozen at minus 20 Celsius degrees. The bee drone larvae harvested and homogenized by the first method were freeze-dried in plastic containers. The total during time was about 24 hours. Most likely the sample was not completely dried because the drying content surface area was relatively small. After freeze drying, the samples in plastic containers began to deteriorate rapidly within a few days. The second method used to harvest the drone larvae was more convenient. At first, empty spaces were left in the lower part of the bee frames. A bee's wax foundation is placed in the upper part where the bees later place the honey. After a while, the working bees formed the bee drone cells in the empty space at the bottom of the frame. The bee drone cells combs are then cut out the frame and frozen in a freezer at minus 20 degrees Celsius. The bee drone larvae are taken, knocked out of the frozen cell combs. The process is similar to bee bread harvesting technology, and the obtained larvae were contaminated with wax particles. For convenience, the larvae were also freeze-dried together with waxes. After freeze-drying, the waxes are easier to remove as it can be done at room temperature. Various methods were used for microbiological and chemical analysis as shown in the table. Drone larvae contain a lot of protein. According to the regulation EC number 1924 2006, it can be claimed that a fresh drone larva product should be labeled as a source of protein, as it has a protein content of at least 12%. Oleic acid and palmitic acid are the most common in this product. Oleic acid is also monounsaturated omega-9 fatty acid. Microbiological analysis were performed on 3 samples. Sample A was harvested by tweezers, homogenized and frozen at a temperature of minus 20 degrees Celsius. Sample B and C were harvested together with cell combs and then frozen. After freeze drying, microorganisms rapidly increased in a sample A. The result table shows that sample A contains much more microorganisms than sample B and C. In contrast, the number of molds and yeast in sample A was lower. Sample A was not completely dried and therefore deteriorated rapidly. Freeze drying in plastic containers proved to be inefficient, as the area to be dried is significantly reduced, which would significantly increase the curing time and the cost and the product. Mucora sarcoineloids and Candida palmata were the most harmful bacteria that were identified in drone larvae, but these bacteria rarely cause infections in humans. The harvesting, processing, and storage of bee drone larvae are impaired by harmful microorganisms such as mucor sarcoineloids and the Candida palmata. Different types of fungi species of the genus Penicillium indicates foliage in the freeze-dried product. It's more efficient to freeze-dry intact larvae because they have much larger surface area to be freeze-dried than pressed and homogenized larvae. A change in color tone of the drone larvae indicates deteriorating during harvesting and storing. Future research is needed into the deterioration of bee drone larvae and the growth of microorganisms during harvesting and storage. The technology for harvesting bee drone larvae is similar to harvesting bee bread when the cell combs are frozen with the product and then separated. Thank you for your attention.
Yes, question. Maybe other participants just concentrate on Yvette's work. Thank you very much, Yvette, for presentation. But I have one question and also one recommendation. How do you explain the high palmitic acid concentration comparing to the reference intervals? You have twicely high concentration.
Reference intervals, it was from literature.
Yeah, it's not more than 1.9 and you have 3.1 and it seems to me twicely high.
So
How do you think? It depends from the feed or it's some genetical thing? Okay, don't worry. It means that you have time to try the answer on this question during the study. But if we talk about the recommendations, actually, I noticed that you have very high yeasts and malts concentration.
Yes.
And this concentration is really very high. Of course, we know that some yeasts and mostly malts are able to produce some toxic compounds, and of course we need to take into account and we need to care. It means that you just solve the question how to improve these hygienic conditions from this raw material till this final product.
Thank you very much.
Thank you, and please continue your activities with energy as you have. Thank you.
Thank you very much.
Thank you. Any more questions? No, I'm guessing there are not any. So thank you, Yvette, for your presentation. And then I guess I will move on with the next presenter, which in this case will be me. So good morning once again. My name is Lian Wozel, and today I will be speaking about evaluation of texture-modified product nutritional composition. First of all, texture-modified products are a part of products for special medical purposes, which is a specific group of products that is a part of a recovery process for people with disabilities. Intake of quality and nutritious food plays an important role in the human recovery, as it provides the body with everything it needs to function optimally. The need for such products can have a variety of reasons, including neurological, neurodegenerative diseases, but also physical trauma, general aging, and so on. In Europe, about 23% of elderly living independently, and up to 51% of in stationary patients, and as many as 84% of those with some form of dementia or a neurodegenerative disease experience dysphagia. Dysphagia is a difficulty of swallowing, hence the inability to swallow food and drinks properly, which this can be extremely dangerous because this can lead to suffocation. For medical purposes, two main types of feeding are distinguished: parenteral and enteral feeding. And enteral feeding includes special oral diet products and enteral tube feeding products. And today I will focus more on the oral nutritional supplements. The commercially available food for special medical purposes in Latvia is uniform, and its range is not very wide because the local market is extremely small. Commercially available products are an internal part of nutritional therapies, but in recent years, medical staff as well as the general public have become concerned about the effectiveness of the use of synthetic nutrients compared to natural sources. Therefore, multi-section research was developed to create new products for special medical purposes. And today I will shortly talk about the part of the research where its main objective was to replicate under industrial conditions laboratorically prepared texture-modified products and evaluate their nutritional composition. So the experimental research was conducted in the laboratories of the Faculty of Food and Technology of the Latvia University of Life Sciences and Technologies. And also in the production facilities of Decay. Some analyses were conducted in also Institute of Biology of University of Latvia, and Food Safety and Animal Health and the Environment Research Institute, BIOR. For this research, a total of 9 recipes were created for this part of the studies 5 desserts abbreviated with D1 to D5, and 4 main dishes or soups abbreviated S6 to S9. The recipes are shown in the table and mostly containing locally grown fruit and vegetable purees and juices. In addition, each recipe was fortified with whey isolate as the main protein source and vitamin and mineral premix to ensure needed daily intake and additional seasonings for sensory purposes. The recipes were fortified with vitamin and mineral premixes due to previous research where it showed that most of the recipes do not contain the recommended daily intakes of several vitamins and minerals. The experimental scheme shows the overall preparation of the product under industrial condition, where after weighing and mixing the ingredients, they were vacuum cooked. Afterwards, additional vitamins and mineral compound premix was added and mixed in well. After that, the product was homogenized, filled in 200 gram stand-up pouch packs and sterilized. And in the end, analyzed based on its dietary fiber content, protein content, fat content, energy value, vitamin and mineral composition. So regarding the results, the total fiber, protein and fat content of the products was determined, as well as the energy value was calculated. There are both positive and negative differences between these products and those already commercially available. Thus, for example, newly developed products have a significantly higher fiber content, and on average, commercially available products do not contain any dietary fiber, which is, however, dietary fiber is extremely crucial for bowel movements. The protein content is also considered to meet the needs of particular consumer group, which is prepared, which in prepared samples varied from 8 to 13.8 grams per 100 grams of product. Commercially available products on average contain 12 grams per 120 milliliters of product. However, the energy value of the products is slightly lower than needed. On average, the commercially available products contain up to 300 kilocalories per 100 grams of product. In these prepared samples, on average, they contained 160 kilocalories per product, with the exception of samples D5 and S9, which where the energy value was close to 200 kilocalories per 100 grams of sample. In this table, the vitamin content of prepared products is shown. As you can see, all of the samples of the products exceed the maximum levels of recommended B12, B6, B9, and D3 vitamins. Vitamin E was in the recommended zone appropriately. It wasn't too high, it wasn't also too low. Regarding the mineral compound content, most of the products contain appropriate amount of recommended amounts, although slightly increased amounts of zinc, iron, and calcium were detected in most products, and decreased levels of sodium were mostly found in dessert samples abbreviated with D. Here you can see this first section, which were not, in these samples, no salt was added, therefore also, which is the suspected reason why the sodium levels were much lower than in soups. So overall, it could be concluded that texture-modified products developed during the study have an average energy value of 160 kilocalories per 100 grams of product, high content of protein and dietary fibers, but their mineral compound and vitamin content partially complies with regulatory requirements. Therefore, it is considered to be suitable as a diet supplementary product, but may not be the only diet for patients with dysphagia. And also by supplementing the developed texture modified product recipes with a premix of vitamins and minerals, it is possible to significantly increase the content of vitamins and minerals in the product. Thank you everyone for your attention. So if you have questions, I'll be happy to answer to them.
I have only one question. You have really very high concentration of zinc in your textured foods. Do you have explanation why you have such a zinc amount?
I believe that it's quite high. Yeah. So I believe this might be a cause of the added mineral premixes that we used, which we ordered from a facility. And I think for future requirements, we should really look into the amount that we ask from them to be added into this mixture, and it definitely needs to be lowered. Yes. I believe that's because from the previous research, I believe these samples did not have this high zinc content. So I think the premix is the reason why it's so high at the moment.
No, I'm just specifying because you said that it's not only food, it's just a food supplement, and then you also recommend another food products in the daily nutrition. Therefore, if we just calculate total amount of some minerals, finally we have quite high results. And in this case, we talk about the overdoses and taking into account this patient's auditorium. We need to maintain this potential stress as well. Thank you very much for your presentation. Great.
Thank you. Any more questions? If not, then I don't mind moving on. So I would like to present our next speaker, which is Svetlana Alexeyeva. Svetlana, are you here?
My presentation is about dairy phospholipids in human nutrition, and I'm really happy to present it here in a great conference. The essential components of the milk-fat global membrane are polar lipids and sphingolipids. Polar lipids and sphingolipids are amphiolic molecules, and they consist of hydrophobic tail and hydrophilic head. Also, glycerophospholipids and phosphosphingolipids represent main parts of phospholipids, but representatives of sphingolipids are sphingomyelin and ceramides. The essential part of a balanced and healthy diet is dairy products, as intake of dietary phospholipids from dairy products varies and approximately are from 2 to 8 grams per day, and it's about from 1 to 10% of the total daily fat intake. Primary dietary phospholipid sources are usual eggs and cereal grains, oilseeds, fish, beef, and of course, dairy products. Dairy products represent a significant source of nutritional phospholipids in the human diet. And if we are talking about the consumption of dairy products in Europe, so it corresponds to about 9% of dietary energy supply. There are different phospholipid amounts in different dairy products. So as you can see, the different numbers for whole milk, cream, butter, skimmed milk, yogurt, and also buttermilk. And as you can see from the presented numbers, buttermilk has highest phospholipid content comparing to other presented here dairy products. And the production process explains these differences, why the difference is present. The chemical composition of milk depends on several factors, and those are environmental factors, feed composition, animal status, lactation period, and season. So there are so many factors that influence the composition. Materials and methods which were used in this research were searching the Web Science Scopus Wiley online journal databases. Time period was searched from 2000 year to this year. I looked through full text papers, and the main keywords were like dairy phospholipids, milk fat, globular membrane, and so on. The main research area were identified, and those are 5 areas. And the first one is that the content of dietary phospholipids and sphingolipids in different types of different products. The second one, phospholipid impact on lowering the cardiovascular disease risk and lipid profile changes. The third one, changes in gut microbiota and intestinal barrier functions. The fourth, phospholipids effect on structural and functional development of the neonatal brain. And the last one, changes in cognitive performance during distress. The research of bioactive dairy components has very considerable potential in human health mechanism. And the number of studies of phospholipid effect on health is still growing, and research becomes more and more big and includes so many factors in that. Clinical research suggests that dietary phospholipids effectually impact intestinal integrity and gut microbiota, inflammation, lipid profile, and heart and circulatory system, brain development. And let's go through all that 5 areas. The first one, intestinal integrity and gut microbiota are correct maturation of the intestinal barrier is significant for health during all life. Milk lipid fraction supports the proper development and stability of the intestinal barrier by an integrity of intracellular tight junction that control permeability. And the evolution and growth of the human intestinal immune system and microbiota development take approximately 2 years. If we are continuing to talk about intestinal integrity, so dairy lipid fractions improved in vitro measures the integrity of the small intestinal epithelial functions. And Gangla diet had 2 positive health promotion mechanisms, and the presence of gangliosides may cause the improvement in burial integrity and response fractures. Phospholipids interaction with bifidobacteria and lactic acid bacteria goes through interactions through phase proteins. And phospholipids enhance the efficiency of probiotic of the gut by promoting probiotic survival and blood immune development. The next area is related to lipid profile and cardiovascular diseases, which are presented by cerebrovascular disease, rheumatic heart disease, coronary heart diseases, and are the main cause of this globally. If we are talking about numbers in Latvia, so about 55% of all deaths were related to cardiovascular diseases in 2019. The significant correlation is present between increases in triglycerides, total cholesterol, and the risk of cardiovascular diseases. So the main role of low-density lipoprotein cholesterol in the pathogenesis of cardiovascular disease and in the lowering low-density lipoprotein cholesterol in higher-risk patients have been demonstrated in very many studies. And thus we can say that the diet plays a significant role in cardiovascular disease lowering and prevention. In plasma and cholesterol homeostasis, low density lipoprotein concentration is supported by intestinal absorption. And in vitro studies has shown that buttermilk, as a great presenter of phospholipid source in dairy products, contains a high concentration and inhibit micellar solubility. Studies show that short-chain consumption of buttermilk significantly reduced triglyceride and total cholesterol levels in women. And the reduction of those parameters was even noticeable among individuals with higher low-density lipoprotein concentrations. Dietary cholesterols, that's cholesterol we intake from the food, increase total cholesterol and low-density lipoproteins. And if we are talking about buttermilk, this is a great and excellent dietary source of sphingolipids and contains about 17 to 35% of total phospholipids. If we are comparing with another source of food of phospholipids, so we can name also eggs. But eggs are contains very concentrated dietary cholesterol. So a study showed that 12-week egg consumption significantly rises total cholesterol and low density lipoprotein concentration in women. Consumption of buttermilk increased total cholesterol and low density lipoprotein moderately. So buttermilk components influence the bioactivity of dietary egg cholesterol absorption and has beneficial effect on health. The next area, cognitive performance during the stress. So we can say definitely that stress reduces cognitive performance, cognition, and often underlies cognitive deficits. And phospholipids have a great lowering effect on the activity and reactivity of the hypothalamic pituitary axis. Supplementation with phospholipids has a positive lowering impact on the stress response. Intake of milk phospholipid has shown the improvement of cognitive performance under high physiological stress. Studies show that...
Svetlana, your time has come to an end. Could you maybe summarize your research?
Okay. So if we conclude the review, so dairy products are a valuable source of phospholipids and an essential part of a human healthy diet. The unique composition of phospholipids provides high nutraceutical properties. Intake of phospholipids positively impacts on the number of health states, and but unfortunately, the positive effects of phospholipids from milk fat globular membrane are mainly based on in vitro animal studies, but there are small studies with volunteers. Thank you very much for your attention. Questions, please?
Okay, then I will ask maybe a question. Why this subject? What are your future goals for this research? Why have you chosen this?
Mm-hmm. I am a first year doctoral student, and my doctoral work will be related to evaluation of buttermilk functional properties. So we will definitely we'll make research of buttermilk, of component of all the main phospholipids and sphingolipids, compare with representatives of other dairy products, and sure we'll make a study on the volunteers to have a look what can we see with results in lowering triglyceride and total cholesterol and all other elements that are influence cardiovascular diseases risk.
Okay, thank you very much. Anyone else? A question? A comment maybe?
Okay, thank you very much, Svetlana, for your presentation. If we analyze this dietary lipids or polar lipids, and then we have a lot of varieties. Do you have clear vision which of lipids are more promising for nutritional benefits to human health?
I think that single lipids and polar lipids, so they are like great sources that can positively affect on the health.
Mm-hmm. And can you say some compounds from these sphingolipids, because the sphingolipids are the group of lipids? Do you have some examples from these sphingolipids which can you recommend for the nutrition? Just examination, okay. You can say that all sphingolipids are very welcome, but of course you will try to understand more about it, because in this field we haven't researches in our country, and still information about these polar lipids are not very clear, because we have a lot of identification methods, we have a lot of compounds, and of course the study in this field is really very welcome. Thank you.
Thank you.
Okay, if there are no more questions, thank you for your participation, and good luck with your future research and PhD work. So next one, I would like to introduce Elisabetta Andersson Tresina and her review on use of peas and beans in high moisture food extrusion.
So good morning, dear conference organizers and participants. As already Liana mentioned, I am Elisabetta, and my supervisor is Professor Tatjana Kinca. And today I will introduce you with the main things that I have been find out in the literature about this theme. This review has been done with acknowledgement in the framework of the project Legumes as Alternative for Extruded Pasta Obtaining, funded and implemented by the program Strengthening of Scientific Capacity at LLU. So the consumption of legumes in Europe is about 7.5 grams per capita per day, but in the comparison in the world, it's about 21 grams per day. Pulse's consumption pattern, of course, is affected by many factors, for example, socio-cultural, economical, and also historical. Improving pulses production, consumption, and usage is important for prevention of malnutrition, micronutrient deficiencies, and also overweight. In the last decade, there has been a scientific interest in extrusion process. Extrusion process can be low moisture and high moisture. The latest trend in food extrusion is high moisture extrusion, when moisture content of raw material is more than 40%, and it seems to have products with better texture and with lower energy input than using extrusion cooking. The aim of the present review was to investigate the latest findings about peas and beans, flour suitability for high moisture food extrusion. Scientific databases such as Scopus, Science Direct, and Web of Science were studied. And the main purpose to summarize and analyze more recent findings about peas and beans, and to focus on chemical composition, functional properties, possibility of pulse flour blend using in high moisture food extrusion, and the monographic method was used. The chemical composition. So the slide is separated into two parts. In the first slide, then first part, so the field B. Of course, if we look at the pulses, we have to look at the starch content and protein content. And in 2020 has been published results from study which was carried out in Latvia, where the main goal was to gather 5-year investigation results of the total protein content, amino acid profile, and total lipid content in different pea varieties, and to estimate their usage for food productions. And here you can see there are a bit differences if we grow peas in conventional system or in organic system, and also there are differences between this 5-year period. If we look at the essential amino acid amount. Of course, we have to highlight the lysine content, and essential amino acid approximately accounted for 35%. And tryptophan is limiting amino acid for yellow split peas. If we look on the beans, of course, again, the starch content and also the protein content. And in the beans, the amino acid score for methionine and cysteine were limiting for red kidney beans and black beans. And the PDCAS is a protein evaluation method combining amino acid requirements of human and the ability to digest it. And here are the results from study, and they look at the cooked beans. And as you can see, the highest results has the navy beans and also the split beans. Of course, we have to talk about starch and the microscopic examination also again of the cooked whole legumes, green, yellow peas, navy beans, and pinto beans explained that a large part of the starch was partially gelatinized and located in the walls of intact cells, while cooked flour pastas contained few gelatinized granules. Processed biological components are anti-nutritional factors. Trypsin inhibitors can reduce specific amino acid availability. For example, they can bind with lysine and arginine. Anti-nutritional factor reducing methods are germination, boiling, soaking, and extrusion. And the literature, I found out that the high treatment temperature as 148 degrees per Celsius and short extrusion time, the moisture content of the raw material was 25%, and the screw speed rotation was quite fast. 100 rotations per minute was the most effective circumstances for tannin, trypsin, and alpha amylase inhibitors reduction in peas without affecting protein content, how it eventuates by germination, dehulling, and soaking. And phenol antioxidant index, this one, which is indicator to sum up antioxidant phenol concentration and their effectiveness. As you can see, the best results was in beans in the cream color and also in red color. But in comparison, I found out in the literature that there is another study where researchers find out that the anti-radical activity of pulse variety was closely correlated with the color of the seed coat. So this is very interesting a place where to look out. Functional properties are very important in new product production. Water absorption capacity. So here you can see the results for yellow peas and gray peas and white beans, also Phaceolus vulgaris. They are quite common. Boiling, roasting, and germination, two types, 24 hours and 48 hours, were used as a flour treatment methods, and all methods increased water absorption capacity among 1.5 and 3 times comparing with untreated pulses. Germination and boiling reduces emulsion activity in white bean flour of approximately 17%, emulsion stability by 25%, but did not change the emulsion properties of gray and yellow pea flours. Germination decreased emulsion activity by 15 to 33% in the gray pea flour and emulsion stability by 14 to 48 in gray pea and white bean flowers in comparison with untreated flowers. And in another study, emulsion activity of pulse flowers contrasted. Small red bean flower had the highest value, more than 92%, and lima bean flower was the lowest value. In this concrete study, it was approximately 63%. Viscosity, one of the most important quality factors in food products, it describes by pasting temperatures. As measurement unit for viscosity is used rapid visco analyzer units, and the highest peak viscosity has black-eyed bean flour. Here you can see to more than 200, and the lowest had a small red bean, and even two times less, 103. If we talk about the peas, then I find out the research when they look at the gelatinization temperatures of the field pea flour, and the peak high index was 0.68, and the pasting temperature was moderate to be 73 to 75 degrees per Celsius. Pulses in a high moisture extrusion. So once again, extrusion at high moisture level above 40%, also known as a wet extrusion. Moisture pressure, shear stress conditions, and temperature, plus raw materials are forced through the heating barrel mixed between screws shaped, cooled, and sheared. Influence proteins cause their denaturation, starch gelatinization and degradation, lipids oxidation, antinutrient trace minerals, and vitamins degradation. Yes, and the current trend in high moisture extrusion is meat analogs, usually made from pea and bean protein powders and isolates, soy protein isolates, maize and wheat starch, microalgae, and peanut proteins or isolates. High moisture food extrusion process using single screw extruder, twin extruder, or shield cell. Moisture content of raw materials were 55 to even 72%. The maximum extrusion temperature is 95 to 170 degrees per Celsius, again, depending on nature of raw material. Weaknesses of technology, high cost equipment, inflexible restrictions on raw material, more than 60% of protein, and moisture content in range 40 to 80%. The most important aspect that the high moisture extrusion is the future development direction. And in scientific literature, main research data is available regarding to soy protein isolation extrusion at 56% moisture using twin screw extruder, or yellow pea protein isolate or concentrate at moisture content in range 66 to 70% using co-rotating twin screw extruder or both combination at moisture content 50 to 70%. Yes, this was that. And conclusions, pulses are a good source of protein, carbohydrates, trace minerals, and vitamins. Chemical composition, functional properties of yellow peas in different varieties of beans, except gray peas, are well described in scientific literature. Germination, especially 48 hours, can be promising pre-treatment for pulse seeds. It increases also folic acid content, water absorption capacity, and emulsion activity. Extrusion technology increases end product protein digestibility and induces changes in antinutrient activity. High moisture food extrusion mostly is used protein concentrate and isolates from pulses, and further research is needed to clarify the possibilities of peas and beans flour blend in high moisture extrusion. And thank you. Any questions?
Hello to everybody. Thank you for your presentation. I have a question about germination. You said that during germination, the protein concentration decrease. Can you explain why it's happened and if this decrease is significant?
No. This 48-hour germination in that study, they found out that the germination is that method which isn't decreasing protein content.
Because maybe it's my mistake. I saw in your slides that protein concentration decreased during germination.
Then maybe this sentence was no. So
okay.
Boiling, soaking, they decrease the protein content, but no, not the germination. No.
Okay, and one more question about you mentioned about these anti-nutrients, but how do you think? What about these pulses and aroma of pulses? How you plan to solve this problem? Because I think it's one problem as well, because consumers don't like or don't prefer this aroma.
Mm-hmm. Thank you.
Do you have any idea how to decrease this negative?
There is aroma, and also the taste. This beanie taste. Also, consumers are not. Don't like a lot. I thank you for a question. I have to look at this and read. I found out one study, but I'm not sure. I'm not. Do I understand right? And I don't want to right now tell some kind of something wrong. But thank you. I have to take a look at this also. Thank you.
Thank you.
Okay, thank you. Any more questions?
Yes,
if not,
yes, if I can, I will ask. You mentioned such features that anti-radical activity of pulse varieties was closely correlated with seed coat color. Yes. What do you think? What is your opinion about this collaboration or coloration? Why this anti-radical activity is higher in colored seed coats?
Mm-hmm. Thank you for your question. I understand. Actually, I took this from the literature. That's why I said these data are very it's quite you can discuss about this question, because in one study they said that there is this correlation, and in some study they say that there isn't.
Maybe it's because of content of carotenes and pigments.
Yes.
Okay. Thank you. And there one more question about extrusion process.
Yeah.
You mentioned that you will extrude these high moisture content as starting material. Yes, it's more than 40%. And after that, you will get these pasta products, I think, yeah? And pasta is a product of approximately 12% of moisture. How will you get off this moisture content? Because extrusion process is very short in your case.
Air drying, maybe.
Drying.
I think drying.
Okay. Yeah. Have you tried already some experiments?
In my master thesis, I worked with peas. I haven't been working with beans, but in master thesis, I worked with beans, with gray peas, yellow split peas, green split peas, and but then we made four recipes with different kind of moisture content, but the highest amount was 25%, and already there were differences in these pasta strings.
Yeah. Yes.
So I will try. I hope I will success.
Yes, good luck.
Thank
because my PhD was about extrusion process of byproducts.
Ah, that's nice. Okay, thank you.
Okay. Thank you very much.
Thank you. I guess Elizabeth has got a lot of suggestions. Now she has just started working. Good luck, you two, for your future researches. And now I would like to invite Ilva Lignitska to talk about the role of cereal and pulse constituents and processing technology in flake texture development. Ilva, please.
Hello, everyone. My name is Ilva Lignitska, and our research topic is role of cereal and pulse constituents and processing technologies in flake texture development. It is a review article. It is estimated that during the period from 2021 till 2026, breakfast cereal market will have an annual growth rate of 4.78%. Also, demand for foods, high nutrition foods is rising. To increase cereal products such as flake nutritional value and functionality, it is possible to combine cereals with pulses. As literature shows, cereals and pulses have a similar carbohydrate, fat, and B group vitamin content. However, pulses, compared to cereals, pulses have a higher protein, folate, iron, magnesium, and zinc content. It shows that it's beneficial and reasonable to combine and partly replace cereal grains with pulses. To produce appropriate quality flakes, it is needed to have knowledge on the relationship between chemical composition, processing parameters, and texture. Processing can have either a positive or negative impact on the flake texture. Also, fundamental changes can happen at the microstructural and macrostructural levels. Such components as starch, fiber, protein, fat, and moisture content can influence grain product texture. The aim of this study was to characterize the role of cereal and pulse constituents and processing technologies in flake texture development, which could be used in flake production for achieving appropriate hardness and crunchiness of new products. Materials and methods. To characterize cereal and pulse constituent and processing technologies' role in flake texture development, the monographic method was used. The latest information published in research articles and books was analyzed and summarized. The published data was used to compile available knowledge on the impact of such constituents as protein, starch, fat, and fiber, and such processing technologies as steaming, cooking, flaking, drying, toasting, and extrusion on flake texture. In total, 77 research articles and books were analyzed. As for results and discussion, here you can see table where proximate cereal and pulse composition is shown. Numbers in black, in bold, in these colored cells show the highest values, and these orange-brown numbers in bold show the lowest values. As it can be seen, cereals and pulses have a high carbohydrate content. For cereals, it varies from 52.9 till 83.8%, and for pulses, it varies from 35.6 till 64.8%. As for starch, white rice and corn has the highest starch content, which ranges till 75.2% for white rice and 83.2% for corn. Results show that pulses have a lower starch content than cereals. As for dietary fibers, white rice has the lowest content, but Hollis oats and red kidney beans have the highest total dietary fiber content. As previously mentioned, pulses have a higher protein content than cereals. Results show that protein content in pulses varied from 20.4 till 26%, And as for fats, whole oats have the highest fat content, which varies till 12.4%. Range of nutrients vary depending on the variety and place of origin of cereals and pulses. Here in this table, you can see mineral and vitamin content of different cereals and pulses. As it can be seen, whole oats are high in ferrum, magnesium, phosphorus, zinc, and thiamine. And pulses are, as previously also mentioned, pulses are rich in ferrum. Ferrum content in pulses varies from 2.1 till 7.6 milligrams per 100 gram of product. They're rich in magnesium, phosphorus, and also thiamine, vitamin B6, and folate. During processing, physical-chemical properties of components such as starch, proteins, lipids, and cell wall component changes, and they have large effect on the texture of grain products. Food texture and also flake texture can be described as expansion index, bulk density, hardness, crispiness, and crunchiness. As previously you could see, the major component of cereal grains and pulses is starch, and during thermal treatment, starch interacts with lipids and proteins and undergoes structural changes. The process responsible process for quality characteristics of high starch products is gelatinization and retrogradation of starch. Another component, as fiber, can reduce the expansion ratio and increase expanded product density and make it harder. It is possible to use different enzymes to depolymerize grain cell wall polysaccharides through hydrolyze. Proteins, cereals and pulses are rich in proteins, and proteins can encapsulate starch granules, inhibit starch gelatinization and digestion, reduce starch structural order, and inhibit starch retrogradation. As for amylose, it can form complexes with lipids, especially monoacyl glycerides and fatty acids, can significantly change the functionality and properties of starch. The complex formation reduces water solubility of starch, decrease swelling capacity, resulting in gelatinization temperature increase, retarded retrogradation, and reduced susceptibility to enzymatic hydrolysis. Here you can see proximate gelatinization temperature, granule size, and amylase content of different cereal and grain starches. Gelatinization temperature varies for different cereals and pulses. For cereals, it varies from 51 till 86 degrees, and for pulses, it varies from 64 till 85 degrees Celsius degrees. Cereals and pulses also have different starch granule size. Whole oats and white rice have the smallest starch granule size, and rye and kidney bean have the highest starch granule size. As for amylose content, it can be seen that pulses have higher amylose percentage, which varies from 17 till 46%. Here you can see general overview of the main processing steps in the flake production. Here you can see these steps for rolled flakes, toasted flakes, extruded flakes, and puffed cereals. For rolled flakes, it includes steaming, flaking, drying. For toasted flakes, cooking, drying, flaking, and toasting. Extruded flakes, mixing, extrusion, and drying. And for puffed cereals, these steps include cooking, drying, and tempering, flaking, and puffing. Before flaking, grains are exposed to steam. This is to increase temperature and moisture content in grains, and it results in gelatinization of starch granules in the rolling process. Flaking is used to flatten grains. To to produce to produce specific shape flakes, and depending on the thickness, it also impacts flake structure. As for toasting, the starting step is cooking, and the main objective of cooking is the gelatinization or melting of crystalline starch into rubbery state matrix to achieve desired shape and size in the further processing steps, such as flaking and shredding. Drying is an important flake processing step, during which it can be possible to achieve desired moisture content, and if flakes have a higher moisture content, they are less hard, less crispy, and more chewier. Toasting is the final stage for traditionally prepared grain flakes, and its product is moved through heating and drying, and it is important factor for color and flavor development. It is possible to obtain flakes. As for extrusion, during extrusion, it is possible to obtain flakes with desired texture and physical parameters directly of the extruder. In this process, due to expansion and shrinkage, the matrix structural properties are modified and texture of product changes. As for conclusions, data analysis show if composition is known, it is possible to combine different cereals and pulses to provide specific nutrient composition in the developed products. Food texture can be food texture in this research article. Flake texture can be described by expansion index, bulk density, hardness, crispiness, crunchiness, and toughness. Toughness and fundamental changes can happen at microstructural and macrostructural levels during processing. Macronutrients as proteins and lipids, physical chemical properties of starch changes, and they have a large effect on the texture of grain and pulse products. And as it's known, the main nutrient in cereal and pulses is starch, and the main change in high starch products is specific gelatinization and retrogradation behavior of starch, and constituents such as lipids and proteins, as well processing technology and parameters can significantly modify the properties and functionality of starch. Thank you for your attention.
Thank you, Hilda, for your presentation. Very interesting. Any questions, please?
Thank you for presentation. I have one question about your product. Do you plan to combine together like a flour to make flour from the grains and from the pulses?
For now, I'm trying I'm working on traditional flakes, which means that I will be using whole grains and whole pulses, not flour, and the process will include cooking, it's like boiling and steaming, drying, rolling, and toasting. And this time for normal traditional 1 grain flakes, after drying, these flakes are rolled and toasted. But for my occasion, after drying, grains will be mixed in extruder as a mixer, and then this mass will go through the rollers, and then will be dried. It will be as multigrain and pulse flakes, where whole grain and pulses are used.
Okay, because for me it was not clear why you analyze these clusterization temperatures and other parameters. If you don't plan to combine them together, what is the main idea of this?
I'm planning. Yes, I'm planning to combine them together.
Yes.
And yes, and it's. I'm looking on this process as also cooking, drying, and tempering, because also in these while I was searching for this literature for this article, it can be seen that I need to know this gelatinization temperature, because during cooking this starch gelatinization happens, and also these grains, cereals, and pulses have different gelatinization temperature, and it means that I need to know I need to find the temperature and these parameters in which I need to do the cooking, drying, and this extrusion temperature to not let the whole mass to have the full gelatinization. Yes. Yeah.
Okay, thank you. And one more question. Have you looked through the amino acid composition and pulses and cereals?
Yes, I've looked through the amino acid and composition in these cereals and pulses, and I've seen that it's possible combining also these pulses and cereals. It is possible to achieve complete proteins, as it's written in this EFSA regulation, which is needed for amino acid content in gram of proteins. Yes.
Yeah, because if we speak about lysine and methionine, we can combine and to get this better proportion of essential amino acids. And I think it's important in your case, because combining you can get this better product with better nutritional properties. Thank you very much.
Thank you.
So thank you, Ilva. So next, I would like to invite Liga Marcinkoniene and her research on fatty acid composition of goat milk for different breeds in organic farming. Liga.
So hello, everyone. And I am Liga, and today my topic is fatty acid composition of goat milk in organic farming. And the aim of this study was to analyze fatty acid profile of goat milk. And goat milk is known as less allergic, with high nutritional value, characterized with high digestibility of milk constituents compared to bovine milk. And fatty acids differ between the animal species. And the fatty acid composition of goat milk triglycerides showed that the highest concentration was caproic, caprylic, and capric acids. And these fatty acids are related to unique flavor and aroma of goat milk. Long-chain fatty acids are considered as unfavorable in human nutrition, and these can affect milk and further cheese quality. Goat milk is rich with fatty acids as butyric, caproic, caprylic, capric, and lauric, myristic, palmitic, and linoleic, but poor with stearic and oleic fatty acids. And it's also known that omega-3 and omega-6 fatty acids are replaceable in human nutrition, and goat milk has higher omega-6 concentration than cow, buffalo, and also sheep milk. In this study was carried out last year from May to July in organic goat farm, the limited liability company Līcīši, and fatty acid profile and goat milk composition were analyzed. 15 individual goat milk samples and 5 bulk milk samples were analyzed for fatty acid composition and fat concentration. And also 27 individual milk samples were collected for goat milk composition, and analyzed breeds were Latvian native, Saanen, and milking crosses, and lactations were from 2nd to 5th. And here we can see the milk composition and quality indicates by goat breeds and lactations. And in our research, the protein and casein concentration, the highest were for milking crosses. But the highest lactose concentration were for Saanen goat breed, and interesting is that also for this breed was the lowest somatic cell count, which is good. And for Latvian native, just urea concentration was the highest. When we, in the many researches, they are also shown that the Saanen goat breed is more productivity in mostly worldwide, and but here we can see that also in our study the milking crosses had good results. Talking about lactations, then we can see that the protein and casein concentration were highest for second lactation, and also the urea concentration was higher for second. And the third lactation had the highest lactose concentration. And for the fourth lactation, the somatic cell count was lowest, which is again interesting because in many scientific papers they are shown that somatic cell count can be higher with higher lactation number. But here we can see that in our case, these results can differ. And also Latvian scientists are established that for Saanen goats and Latvian goats are also higher protein and fat concentration. And here we can see that the fat concentration in our study was higher for milking crossed breeds, which differ from the other results. And here we can see that the Saanen goat breed has the highest butyric and caproic and caprylic acid, and almost all others were higher for milking crossed breeds. But if we compare with different results in literature, then our results was quite low with the fatty acids. And it can be also one of the reasons that in this farm, they don't have so much this special flavor and aroma for goat milk. And that's why it can be because of these fatty acids, caproic, caprylic, and capric is quite low. But for Latvia, it's good because Latvian people like that they are not so high, they're not so special, this aroma and flavor for the goat milk, and also further for the goat cheese. And here we can see some other fatty acids, and here the milking crosses had the higher total saturated fatty acids and total monosaturated fatty acids also. And here the total omega-9 fatty acids was also higher for milking crosses. About omega-6, we had quite low this amount. But it is good for human nutrition to use also goat milk in their daily diet. And also we can see that fatty acids can differ between the breeds, and the highest were fatty acid concentration in milking crossed breeds, and goat breed and lactation affects milk composition. And milk fat, protein, casein, and lactose concentration was the lowest, but the somatic cell count and urea concentration was the highest for Latvian native goat breed. And milk composition, except somatic cell count, were higher for second lactation breed. And also these differences between the different scientific researchers can be because the animal diet can affect this fatty acid amount. And I also found that if there are higher supplements with the fat sources in animal diet, then also these fatty acids can be higher. So research results show that the tendency that fatty acids related to this unique goat milk flavor were high for Saanen and milking crossed breeds. So, and thank you for your attention.
Thank you, Liga, for your presentation. So, any questions?
Thank you for presentation. And I have a question, maybe I didn't notice about this season. Do you pay attention to the season, or it was collected through the year, or you collect samples at the same period of time?
Yes, and thank you for a question. We collected samples in May and July last year. So it was like after the kidding time was over, and then it's like the beginning of the whole season when so it's like
it means that lactation cannot influence these proportions because it's the same season.
Yes.
And one more question about this percentage of fatty acids, because in your case it was grams per 100 gram of product. But we know that there is possible to analyze in percentage the same amino acids, because if we look on your data, you said that the concentration, fat concentration was different in different groups. And of course, if we have high amount of fats, we have high amount of different fatty acids. Maybe it's interesting to compare in this proportion.
Yeah, of course. Yes, this was like the first time with this research with fatty acids. So, and of course there are quite a lot to study more, and I think that we can try to make some different calculations.
And last question: which milk will be the best one? If we speak about fatty acids, what is your advice for us?
Goat milk. Of course, the goat milk is the best for everyone, and we should drink it. And but I think that in our results, quite good results was for milking crosses. But I think that I should see more about this bloodness, these lines, about this milking crosses, because in this farm we have also this like crossbreeds is with Anglo-Nubian goats and Saanen also, and to see exactly which when this change is happening, because these milking crosses, they are like really different breeds inside. So. Thank
you very much. And
okay, thank you, Liga. It's very interesting. Let's all drink goat milk. Lastly, I would like to introduce our final speaker for today, for this section, I'm sorry. I would like to give the word to Evelyn and her research review on reduction of possibilities of gas emission from meat processing companies. So, Evelyn, please.
Good day, dear organizers and participants of the conference. I am first-year student from Latvia University of Life Science, the Technologies Faculty of Food Technology. My theoretical research is connected with education possibilities of gas emissions from meat processing companies. The environmental situation in the world is getting worse every year, and is also gaining momentum in attracting the attention of government agency and prompting adjustment to tighten tax rates on emissions into the atmosphere in order to encourage manufacturers to take measures and introduce practices, as well as to look for methods to reduce emissions. The meat and dairy industry accounts for 14.5% of global greenhouse gases. This type of production contributes to global warming by generating substances that destroy the ozone layer and is also distinguished by its high consumption of water and energy, which provokes increased waste and wastewater discharge. Research aims the purpose of the present research was to consider possible method for reducing emissions into the atmosphere during the heat treatment of meat product during smoking, and to evaluate the effectiveness of method for reducing emissions into the atmosphere with the identification of most effective in achieving an environmentally economic balance, as well as with further installation of meat processing production. The monographic method was used to summarize and analyze the latest information and research articles dedicated to possible emissions reduction techniques. The method of catalyzing smoky smoke using a catalyst system. The smoke catalysis system combines heat treatment and a catalyst with which it safely removes harmful substances from the smoke. And smoke after boring system using high temperatures. Gases are kept at similar temperatures for a long time, as a result of which substances are oxidized with oxygen and destroyed. The chamber is also equipped with automatic removal of combustion ash. Scrubber is an industrial equipment that is used to clean emissions from dust and gases based on wet filtration. The equipment is a column in which the main retention or neutralization of impurities is carried out using water or an absorbent. And Biobed filter system is based on filtration of the gas stream using biological purification. The gas stream passes through biological material. We create a thin water film with microorganisms. Analyzing all four types of possible method and system to reduce harmful emissions into the atmosphere. Each identified its advantages and disadvantages. All the described method relates to difficult solution to the problem on the part of the resources spent. The economic side plays an important role in choosing a suitable method of solving the problem of emissions. The most expensive is a scrubber and a smoke after boring system. But it is also worth considering further investment and maintenance of systems. Conclusions. Catalysis system can be attributed to the optimal choice of a system for solving environmental and economic problems. It has shown a good result of effective filtration to reduce emissions, and is also optimal in terms of maintenance, cost, and dimensions. Biofiltration has the least result. Gas purification is less effective, and there also a need for constant maintenance. But each system is suitable for certain function. It is necessary to consider the result to be reached, the type of production, the composition of gas, the dimension of production, economic opportunities, and campaign policy and location. Reference, and thank you for attention.
Thank you, Evelyn, for your presentation. Very interesting. Does anyone have any questions? Well, maybe if somebody is still thinking, I might have a small question. Maybe in your own opinion, which method would be maybe the best, the one maybe you would suggest, more sufficient maybe, or for any other reason?
I think the better method is catalysis filter, because C is not expensive, and is better reduce emissions.
Okay, thank you very much. Maybe there's a question from someone. Looks like there is none. So then I am happy to say that we've finished today's session on food sciences. Very happy that we've been able to squeeze in the amount of time that we have. Thank you everyone for your presentations, for your participation, and good luck with your future research, and hope I will see you in other conferences and read your research papers. But I will say goodbye from you today for this from this section. And now there will be a short coffee break, and the conference will resume at 12:45. Thank you, everyone.
Okay, hello everybody. Once again, we are continuing our conference today. And yeah, my name is Toms Kondratovic. I will be the chairperson for a session of Forestry. I was offered the honor to do this, so I will try not to delay this any further. Just a quick reminder, each speaker will have 10 minutes and 5 minutes for discussion. So 15 altogether. Let's try to keep it that way. So without further ado, first speaker is Alice Bleve, and she will have presentation on internal decay assessment using drilling resistance in mature common alder stands. Alice, please.
Hello, my name is Alice Bleve, and I work in Latvia State Forest Research Institute, and also I'm a student of master's program of forest sciences in Latvia University of Life Sciences and Technologies. And today my presentation will be on topic internal decay assessment using drilling resistance in mature common alder stands. So let's begin. So why our research is needed and important? Because common alder is very widely distributed tree species in Europe North region. And according to National Forest Inventory data, common alder stands cover approximately 6% of forests in Latvia. Age-related decline of stand biomass accumulation and loss of productivity in old growth forests is very natural and well-described process. And possibility of internal stem diseases, for example, such as wood decay, sufficiently increases after common alder reach its rotation age in Latvia, which is 71 years. Decay presents may reduce not only amount of high quality wood, but also may cause losses of stored carbon in tree biomass, and also negatively affects tree stability. Common alder wood have very high usage potential, so it is important to understand factors contributing decay occurrence in alder stems and also within the tree stem, and to explore new methods of this disease identification also. Because internal stem decay detection using only external tree evaluation is almost impossible. So far, internal stem decay assessment mostly is done by destructive methods, which is tree felling. And the aim of this study was to test the potential of microdrill to non-destructive detection of decay in stems of common alder, and also to assess the occurrence of internal tree decay in common alder stand. And in those pictures, you can see a device which is called resistograph. We use it for our study, and the methodics of this device, how this device works, I will explain later. The presence of internal stem decay by non-destructive device method was studied in 7 mature and naturally regenerated forest stands dominated by common alder located in different parts of Latvia. And in all 7 investigated stands were growing on fertile soils with increased groundwater level. To assess the occurrence of stem decay in forest stands dominated by common alder, a circular sample plot was established in each of it. Also, in each sample plot was measured diameter at breast height and tree height. Average tree age in a stand was determined according to the information from forest inventory. But the other stand characteristics you can see in this table was calculated from the obtained tree measurement data. So, as previously I mentioned, that internal stem decay is almost impossible to be detected only by visual evaluation methods. So, device resistograph is one of the devices which allows to assess stem decay presence, distinguish development stages, and also to determine distribution within cross sections without limiting tree growing. The principle of this micro drill work is to penetrate very fine needle at constant rotation speed and straight line movement and accurately measure the resistance and changes of food density of drilled study object. And in our case, this study object is common alder stems. In established sample plots, all trees were drilled by resistograph at stump level from 3 different directions towards the core. It was done with the aim to make conclusions about stem internal condition as precise as it is possible. And also to verify data obtained from resistography from some of drilled trees with increment borer were also taken wood samples, or those trees were even felled down. Each resistograph drilling has individual graph, or so-called drilling profile, and after quick visual analysis of it, is possible to make assumptions about drilled stem internal condition. Pictures in this slide represents individual drilling profiles in comparison with actual common alder cross cuts. When drilling profile is stable and has no changes, there is intact or so-called healthy wood. Fluctuations may indicate an internal stem decay presence, which cause changes in wood mechanical properties. Those pictures represent 2 easily detectable and well-developed internal stem decay stages. Spongy rot in picture A and cavity in picture B. At these stages, drilling profile line fluctuations are very notable, as you can see. When microdrill needle reaches spongy rot in the tree stem, graph fluctuations become lower and are very unstable. But at cavity, this graph has very significant and stable decrease, which makes very easy to recognize exactly this decay development stage. And after all data collection was calculated mean associations between forest stand parameters and the occurrence of internal wood decay. Also was determined how successful is resistograph to use to assess decay presence in common alder stands. And now we came to the results and conclusions part. 2 wood decay stages, spongy rot and cavity, was successfully detected with drilling resistance method. But by this device, our study revealed very high variation in occurrence of decayed trees in studied nature common alder stands. Spongy rot dominated in all investigated decayed common alder stands, but cavity was found less frequently. As cavity is last stage of internal stem decay development, where wood is no more found, highest occurrence of it was found in oldest study stands and with greater diameter at breast height and respectively mean tree volume. In this table is represented data obtained from work with resistograph. But it is important to mention that during sample tree analysis by increment borer was detected also some wood discoloration. Wood discoloration is determined as very first decay stage, which do not have any significant negative impact on wood density. So in our study was concluded that the presence of discoloration in common alder wood cannot be noticed with this non-destructive device. And those results might lead us think that detecting internal stem decay using only registograph can underestimate the actual amount of decay in forest stands. So, and as you can see, linear regression shows significantly positive correlation between occurrence of decayed trees and stand age. Also, analysis showed positive correlation between decay occurrence and mean tree diameter at breast height and also mean tree volume. Our study also proved initially made assumptions that occurrence of internal stem decay in major common alder stands depend on tree size and stand age. Respectively, the older or larger in diameter is tree, the higher possibility is that it has well-developed internal stem decay. And based on limited data, we consider that internal decay occurrence sufficiently increases after common alder trees reach 60, 70 years. And also one of conclusions is that preserving of old-grown common alder trees induce sufficient loss of high-quality wood due to internal decay, because the biggest proportion of decay is found at the bottom and in the hardwood of tree stem. So if wood industry have interest in common alder wood management, the reduction of actual harvesting age of common alder stands should be considered. And despite the fact that data set obtained from our study is very limited and investigated was only 7 common alder stands, it is already clear that resistograph is very useful device in preliminary internal stem detection in forest stands. And to obtain more accurate data about the internal decay detection and occurrence in common alder stands in Latvia, we recommend to continue this study with more extensive data set and also to try to use resistograph for other tree species. So at this moment, I want to say thank you for your attention. And if you have any questions, I would be very glad to answer them. So thank you.
Okay. Thank you, Alice. Anyone, any questions? Okay, then maybe while others are thinking, I will ask one quick question. Actually, I wanted to ask as I understood you already answered during this presentation. If I understand correctly, you can't use this non-destructive method as a sort of preliminary to determine. Use this method as a precursor to say that rot will occur after something. Did I understood it correctly? That you have to like.
Can you repeat your question? Actually,
basically, that you can't use this method to predict that stem will rot at some point. Like that, there are some places that are looking so to say shady or that will rot at some point.
At this study, the aim of this study was to see how actually useful this resistograph device is just to detect decay presence in stems. But we are continuing this study, and later we will also I don't know, I forgot that word. We will also study the factors which are contributing the decay occurrence and in stems and within the stems. I don't know how to answer differently.
Okay, basically what you mean is that this is like preliminary results and some more. We can assume that some more interesting results will follow. Okay, we have time for one more quick question if somebody wants to ask. Okay, thank you, Alice. If no one is asking, then I will Announce the next presenter. Yes, this next work will be also from Silava, collective of plant physiology laboratory, and I have the honor of presenting these results myself. So as I mentioned, yeah, my name is Toms Kondratovic. I will be presenting results for this study, and our study was about effect of different spectral composition LED lighting on silver birch in vitro plantlet morphology. So in few words about importance of this topic. Maybe many of you know that currently micropropagation as a method for production of superior forest reproductive material is commonly used, and plants in this environment, in these in vitro conditions, are affected by various environmental factors, similarly to outside conditions. And in this case, I would like to emphasize the light quality, which is quite important and can affect plant growth on many levels. And regarding birch, several researches have been made concerning birch propagation itself. However, the impact of this spectral composition of lighting used in this in vitro culture cultivation has not been studied that much. Therefore, aim of this study was to investigate effects of various spectral composition-led luminaries on morphological development of silver birch clone microshoots. To do this, we determined the effect of silver birch microshoots cultivated under 4 different light treatments. For cultivation, we used woody plant mediums supplemented with sugars, plant growth regulators, and solidified with agar. For plant material, we used already established approximately 5-year-old in vitro cultures of 10 local origin birch progenies, which were routinely cultivated in laboratory of plant physiology in Silava. And these transplanted cultures were grown for 6 weeks in climatic chamber where constant environmental conditions were maintained during the experiment. And after 6 weeks, when plantlets had totally regenerated, we determined morphological parameters such as main shoot elongation, total plantlet length of third internode, as well as total and average leaf area. And we also determined fresh and dry weight for these samples. A bit more about light treatments. As you can see, as I mentioned, we used 4 different spectral qualities. First figure on top left is a spectral composition of fluorescent tubes that are commonly used, and in our case were used as control lighting. As you can see, 3 distinctive spikes of emission at blue, green, and yellow-orange region, and this is quite different from all the LEDs we were using in this study. For next treatment, figure B is red and blue LED luminary, which consisted basically of blue and red diode, which are in accordance with plant absorption maximum for photosynthetic reactions. Next spectral composition was red-green-blue luminary, which is basically the same red and blue, but only there was added a bit of green light. And last was the full spectrum luminary, where also yellow and orange spectral parts were added. In reality, it all looked like this. On top, on the far left, you can see this red-blue luminary. Then it's this red-green-blue, then red-green-blue, yellow-orange, and fluorescent light, which were used as control. We used such multi-story shelf systems, which were separated by these screens in order to prevent the effect of neighboring light treatments. What we discoveed was first, as results of linear mixed model show, morphological parameters of birch in vitro cultures were indeed affected by light treatments. However, this response was clone specific. In case of stem morphological characteristics such as main shoot elongation, total plant length, these variables were determined by clone-specific responses to light only, while for length of third internode as well as average and total leaf area, significant direct light effect was also observed in addition to this interaction effect of light and clone. And surprisingly, we did not detect any light effect on stem and leaf fresh and dry weight. And it was only clone dependent. Therefore, I will not try to discuss about this more because our focus was mainly on the effect of light treatment. This is just curious. We were not expecting that these masses won't be affected by light. Yes, due to the effect of clone, we analyzed the results on level of clones. So basically, clone 54229 formed significantly longer main shoots and also had higher total plantlet length and larger total leaf area under fluorescent light, indicating that this clone specifically reacts to lower the red light amount in combination with yellow and orange light, which is emitted by fluorescent tubes. Opposite effect was observed for clone MED36, which had significantly higher total plant length under this full spectrum composition LED luminary, and yes, compared to fluorescent tubes. Basically, what this means, these both clones show response to red light in combination with yellow and orange, but you could say that these responses are opposite. And also we need to take consideration that this red-blue and red-far ratios could be important as well for these two clones. Interaction of light and clone was effect was also present and observed for length of third internode. In case of 4 clones, clone 45286 had a significantly longer third internode under fluorescent when compared to all LED treatments. Partially opposite effect was observed on clone MA29, where length of third internode was of this full spectral LED was more than fluorescent, but did not differ from other LED treatments. Both these clones displayed sensitivity to red light, however, similarly to previously mentioned clones, effect was opposite. And also, column 54299 must be mentioned. It formed significantly longer third internode when grown under fluorescent treatment compared to red-blue and red-green, blue-yellow, which could actually indicate that this blue-green ratio, which triggers shade avoidance strategy in plants, could be related to these differences. And clone MED36 had positive responses to luminaries which contained yellow and orange light. And yes, so longer internodes were observed for all luminaries compared to red-blue LED treatment. And when looked at on this whole species level, not as individual clones, we observed the total leaf area, average leaf area, and length of third internode were significantly smaller for RB treatment, basically indicating that this treatment was underperforming compared to other treatments. And in general, it can be summarized that light spectrum containing blue, green, yellow, orange, red and far red spectral parts is important for the silver birch growth in vitro. For conclusions, we can draw that yes, first of all, these cultures were indeed affected by light spectrum, but this effect was clone specific. Main shoot and total plant length was affected by amount of red light, though this effect can be opposite depending on clone. Clone-specific responses of length of third internode were determined also by red light, but in this case, this effect of red light was present if there was some combination with yellow and orange light. And overall results show that cultures require full spectral composition and will be inhibited by narrow spectral compositions such as RV treatment. Knowledge of specific responses must be taken into account when we speak about propagation of these specific clones. I mean, you have to optimize this propagation process according to each clone. And yes, results show that LED luminaries could replace forests and lights for propagation of silver birch. So thank you for attention. Any questions, please? I will ask to next presenter to come forward, and that is Aldis. Aldis, please, you have 15 minutes.
Yeah, okay. So it's me, Aldis Butlers. I'm from Latvian State Forest Research Institute. This presentation is regarding the study results of the topic case study on the greenhouse gas fluxes from flooded former peat extraction fields in central part of Latvia. In various discussions in Europe, flooding of organic soils is proposed as a climate change mitigation measure. Therefore, it is considered also as a potential measure in Latvia, although there were no studies conducted in Latvia to confirm that flooding reduces emissions. This study was implemented to acquire preliminary knowledge on greenhouse gas emissions from flooded areas of peat extraction. Some background information, total area of organic soils in Latvia is around 20% of the country area, of which close to 5% are former peat extraction sites. More than 5,000 hectares of former peat extraction sites are flooded. So far, to estimate greenhouse gas emissions from such areas in the scope of National Greenhouse Gas Inventory, emission factors for wetted organic soils is applied to avoid the potential underestimation of greenhouse gas emissions. I will get back to those numbers you can see in the slide. In the last slide, when I will compare the study results with those emission factors we use in the inventory. For this study, 3 former raised bogs were selected in central part of Latvia. Peat extraction in these areas was terminated 25 to 30 years ago, leaving around 0.5 to 3 meters deep peat layer in the measurement sites. Water depth in these sites were from around 0.7 to around 2 meters deep during the study, which reduced by up to 30 to 60 centimeters during the summertime. In this and the next slide, we will see study sites. Here we see aerophoto of peat extraction site with the moisture prognosis if artificial groundwater table changes are excluded. Purple color indicates peat will be not saturated, and green shows saturated or periodically flooded areas. Modeling is based on the surface elevation data. It shows that without drainage, these areas would still not be flooded. In this example, we see that without any additional action, few yellow areas will be permanently flooded. This shows that even if flooding will be identified as a climate change mitigation measure, additional irrigation actions will be needed to flood those areas if it's planned to do so. So the next slides will be dedicated to study method and results. In the slide, we see floating chambers in the study sites being installed. We can see installation of food bridges an example how these gas sampling chambers looks like. Food bridges were installed. Food bridges were used to access three permanent sublets established per study site. Gas samples were collected by placing non-transparent chambers on set of 5 floating collars. Gas sampling was conducted from January till December of 2021. Sampling was repeated once per 3 weeks during vegetation season and once per month in November, from November to February. To collect the gas samples, 50 milliliter under pressurized glass bottles were used. First, the gas sample was collected immediately after placing chamber on the collar. Each next sample was taken with a 10 minutes interval. During gas sampling, water and air temperature was also measured, and these collected water samples were transported to laboratory and tested by gas chromatography method. These acquired results of gas concentration and time of measurement were used to calculate greenhouse gas emissions from water surface in time. You can see linear regression here as an example, and preliminary data screening by discarding measurements that does not follow this linear regression were discarded. And as additional measurement, when this linear regression was created, the equations that does not have at least determination coefficient at least 0.7 were discarded from the further evaluation. And the B coefficient of these linear regressions were used by this formula. Basically, it applies ideal gas law, and that way we can calculate emissions from water surface to atmosphere in time. Some results here. During summer months, all fluxes are increasing. And then in the winter time, these emissions are negligible. In graphs, we see relationship between the greenhouse gas emissions and the measurement month. CO2 and methane emissions are here in the upper graphs, and the N2O fluxes we see that are quite negligible as a whole year. Monthly fluctuations of CO2 and methane emissions can be explained by a polynomial equation with moderate coefficient of correlation, but I will not suggest to use these regressions for greenhouse gas estimation prognosis, as these are just a few data points still anyways, as this is a preliminary study. CO2 emissions exceeds uncertainty rate during the fall year, except the period when water is covered with ice. Therefore, in future studies, it is reasonable to measure greenhouse gas fluxes only during ice-free period. Similarly, also see methane emissions exceeds uncertainty range from June to October, and during other months, methane emissions are quite insignificant. Therefore, this source can also be measured only during summer and autumn months. However, between years, different temperature conditions may be present, and that should be taken into account. And as we can see here, again, N2 emissions are insignificant during the fall period. However, in the summer and autumn months, N2 emissions likely exceeds uncertainty range. The acquired results also points out the necessity to increase the regularity of measurements during spring period to catch these greenhouse gas fluxes which occur during ice melting periods, and cracks and openings in ice should also be considered in measurements and calculations of fluxes. Regarding temperature measurement results, we can see that air temperature may be used as a predictor for greenhouse gas emissions, as there is linear correlation between water and air temperature. However, CO2 itself has weak correlation with water temperature, but the methane has moderate correlation with the temperature. This is opposite what I'm used to see in the other studies when we estimate direct soil greenhouse gas emissions. And regarding some exact numbers, According to the study results, flooded areas is a considerable source of CO2 emissions. It's around 1 ton of carbon dioxide carbon per hectare in a year. However, the most significant greenhouse gas in flooded areas is methane. It's around 400 kilograms of methane carbon per hectare in a year. When we compare these acquired results with the, when I mentioned previously, the emission factors we currently use, we can see that it's around twice more than we currently are calculating, and we thought that these are like we tried basically to calculate, not to think that this flooding is like a good thing. It turns out it actually even worse, and we tried to be like at least calculate something. Let's say basically shows that these results are quite similar regarding at least methane emissions, like to drainage ditches. It's quite logic actually. So it clearly shows that flooding is not such a good climate change mitigation measure as some people think. Some final conclusions: estimated net greenhouse gas emissions from flooded areas are in CO2 equivalents 80 tons of CO2 per hectare in a year, which is about twice than currently accounted greenhouse gas emissions in national greenhouse gas inventory. And the most considerable source is methane. And CO2 and methane fluxes are correlating with the water temperature, especially methane emissions, which are occurring only if water temperature is above 5 degrees. CO2 emissions continues also during winter months if water surface is not covered with ice. Flooding of peatlands cannot be recommended as a climate change mitigation measure before more detailed studies are done. And further studies are necessary to increase accuracy of these elaborated emission results, as well as to improve activity data that is necessary to calculate these emissions in the national greenhouse gas inventory, to know how many areas of the flooded areas are and what is the temperature of the water during the year and so on. That's it from me. Thank you. Do you have any questions?
Yes, thanks, Aldis. Any questions? Okay, while others are thinking, I wanted to ask you one question. Can you go to slide before conclusions?
Before?
Yes, this one. Did I understand correctly? CO2 emissions were I assume you compared these results to some sort of reference with dry peat areas. Were these results higher for CO2 emissions or lower or the same, more or less?
For drained peat, you mean?
Yeah. Well, in this study compared to drained peat, yes.
Drained peat has CO2 up to around 8 tons per hectare in a year.
Oh, okay. So this was lower.
Yes, significantly.
Oh, okay, yeah, yeah. Okay, I was just getting confused because I thought you were telling that. Well, yeah, never mind.
The main problem is methane. It's in CO2 equivalents. It's 25 more than CO2 impact on the climate. So it's the main problem.
So basically, if you have to choose, you can live with increased CO2 emissions, but it's better to reduce methane.
Yeah, it depends from the total budget. It's not that simple, but yeah, it's always about this balance.
Yeah. Okay, thank you. Any other ones to ask some questions? Quite interesting presentation. I was always wanting to ask something about this. Okay, if nobody wants to ask anything, then thank you, Aldis, for interesting presentation. And I will ask the next speaker, which is Inga Straupe, to tell about her work on sport activities' effect on Venus forest areas. Inga, please, ground is yours.
I'm very glad to participate in this conference and to deal with you our research. And the research is about the reaction of Scots pine forest ground cover vegetation to orienteering competitions in the seaside Latvia. And I am from the University of Life Science and Technologies, and my master student Ieva Ivbule now is working in Silava, and she is very interested in this orienteering sport. I could say that orienteering is the most popular folk sport in Latvia. And for example, in 2019, more than 16,000 participants took part in competitions. And always when we choose the place where to organize orienteering competitions, we must follow the guidelines of this organization process prepared by Nature Protection Board and the Latvian Orienteering Federation. And this organization of orienteering competitions in specially protected nature territories is regulated by several regulations of the Republic of Latvia. And orienteering has an impact on nature and its values. For example, disturbance of mammals, trampling of vegetation. They are very important factors during orienteering competitions. Especially the edaphic changes in the soil, such as changes in soil moisture, erosion, and structure, can also occur in connection with trampling of vegetation. The aim of our study is to assess the impact of orienteering competitions on ground vegetation at the seaside. And in order to achieve this aim, the following tasks have been set: to analyze the vegetation around the orienteering control points before and after the competition, and to compare and evaluate the changes of vegetation on hilltops and depressions and finish checkpoints. And we chose two research objects, and they were selected because here were big competitions, World Masters Orienteering Championships 2019, and these territories were situated in the seaside near Bumbukauns and Garezeri. And the collection of data took place in July 2019, before and after the competition, and in totally 10 control point locations were selected, 5 in Bumbukauns, 5 in Garezeri, and vegetation was counted in each sample plot using Brown-Blanquet method. And in each sample plot, the total projected cover in percentage of vegetation was determined in each layer in tree, shrub, herb, and moss layer. And the intensity of the trampling depends on the number of participants crossing the sample plot to check in the required control point. And two-factor analysis of variance was used for mathematical data processing. And here you can see the first table, first 5 control points in Bumbukauns. And here 2 control points were in depressions, 2 were on hilltops, and the last one is a finish checkpoint. And here is the cover of vegetation by layers and before and after competition. And you can see that no changes in the tree layer, shrub layer, and herb layer. Only we can observe changes decreasing percentage of cover of mosses in moss layer. Here you can see some examples in pictures. For example, the first picture is in the depression. This control point is in depression. Here the control point is on hilltop, and the last one is checkpoint, finish checkpoint. And in all pictures and in the nature, we observed that the center of these control points are trampled, but distance 7 meters and 14 meters, there are practically no visual changes. Very similar situation is in the competition area in Garazari. Here is the same table, other figures, and here you can see the two control points in depressions, control points on hilltops, and one finish checkpoint. And the same situation. We can observe only changes in the moss layer, and the percentage, cover percentage decreased. The situation in pictures. Here is not only forest type Cladinosa or Colonosa, but also Vaccinosa, but at the same time the changes we can observe only in moss layer. And it's very characteristic for all control points that the ground is trampled mostly in the center. And for the sample plots in the hills, the center is more affected than in the depressions. But the effect of trampling on flat terrain is small or practically non-existent, and the differences are not significant. The participants visited the control points in the hills from one side of the hill, one slope of the hill is more trampled, but the depressions, there are several trajectories from which the control point was visited. The intensity of trampling varies depending on the number of participants, but there are no regularities. The impact does not only depend on the number of participants, both the diversity of ground cover plants and the characteristics of the terrain and soil must be taken into account. And conclusions. The effect of trampling is observed only in the moss layer. At control points, the projective coverage of the moss layer has decreased by 1.8% on average. On other layers of the forest stand, there has been observed no impact of trampling. The most pronounced damage to the forest floor can be detected at the control point itself in the center. As the distance from the control point increases, the trampling intensity decreases. There was no correlation between the terrain form at the checkpoint, the amount of participants, and the intensity of trampling. The impact depends on both diversity of ground cover plants and the terrain as well as soil properties. This research was supported by the grant of project of Latvia University of Life Science and Technologies implementation of research program. Thank you for attention.
Thank you, Inga. Interesting presentation. So if everyone has any questions, please. Okay, I wanted to ask about this. You mentioned that, if I understood correctly, in places in places where the ground was flat, this trampling effect was not so distinct or even non-existent. So based on this, did you do any recommendations for these organizers of such competitions, or for this law making process, so to say, well, basically, did this work result in some sort of recommendations?
I would say that it's not possible to give some proposals for this law or for different these guidelines, because we have not so much research in this field, but we can widen this research and to make the similar research in more fertile places, because at the seaside it's very pure soils, very pure forest stands, Cladinosa, Calunosa or Vaccinosa, but maybe the situation is another in the more fertile places in the forest. But at the same time, we know that the most fertile places are not so sensitive to trampling, yes, but these poor places as Cladinosa, Calunosa or Vaccinosa, they are they are very sensitive to trampling. Yes, but we need more research, and we need to put together our observations, and then we can give some proposals maybe for organizing of these competitions. Thank you for your question.
Yeah, well, yes, thank you. That was actually precisely what I wanted to hear. So basically, with more research, you had the data, yes, to so to say, have background for recommendations. I don't know, for example, for regarding conservation issues or such things. Basically, I mean, these seaside territories are, after all, yeah, as you said, quite sensitive. So yeah.
Mm-hmm. Thank you.
Yeah, thank you. Okay, any more questions from other speakers, listeners? Okay, if not, then thank you, Inga. And I will ask the next speaker, which is Santa Kaleja, and she will also have presentation regarding peatlands and mitigation processes.
Hello. Well, my name is Arta Bārdu, and I'm from Latvian State Forest Research Institute Silva. And this presentation will summarize the main findings of review of climate change mitigation measures applicable in degraded peatland in Latvia. The aim of the study was to identify and evaluate climate change mitigation measures applicable in wetland, including identification of missing information necessary to quantify the mitigation potential. In Latvia, degraded peatlands typically have complex land use structure. And for instance, in this example, active peat extraction fields, shrub cover, forest cover, flooded area, bare ground, and grassland is presented all together. There you can see simplified schematic comparison of GHG emissions in undrained and undrained peatland. Red arrows show methane and CO2 emissions, as well as dissolved organic carbon leaching, and green arrows show methane and CO2 removals. In general, drainage reduce emissions of methane and increase emissions of CO2, as well as increase leaching of dissolved organic carbon. But what is important is to evaluate netto GHG emissions and CO2 removals from all ecosystems, including increase of CO2 removals in tree biomass through photosynthesis processes after drainage. The dynamic of carbon balance and GHG fluxes in peatlands are highly dependent on peat hydrology, respectively on groundwater level and subsequently on oxygen level in soil or soil aeration. And under natural conditions, peatland are growing ecosystems with water table close to the soil surface, as it is seen in this graph for transitional and raised bogs. Peatland drainage causes immediate lowering of water table level, and it subsequently increases availability of oxygen in upper soil layers. Groundwater level tends to fluctuate among seasons, and thus also GHG emissions are dynamic and differ among seasons. And in additional to groundwater level, important factor influencing dynamic of GHG emission is soil and air temperature. There are GHG emission factors from differently managed peatlands, such as grassland, cropland, peat extraction fields, forest land, as well as from pristine raised and transitional bogs. And emission factors here show some of CO2, methane, and N2O emissions. And the highest emission factor was found for transitional bog and followed by research sites in cropland where cereals and legumes were cultivated. These results were obtained within International Life Restore project. So what land use types are dominant for organic soils in Latvia? We have 41% forests, 40% wetlands, around 10% cropland, 5% grassland, 3% peat extraction areas, and below 1% flooded and rewetted wetlands. And total area of degraded peatlands is around 40 kilohectares. To identify climate change mitigation potential measures, we did literature studies, as well as we use expert questionnaire. And in total, 7 expert teams from Latvia, Lithuania, Estonia, Finland, and Germany were involved in completion of questionnaire. And most of experts providing answers are involved in GHG inventory, so they are highly competent. As a result, we obtained a list of climate change mitigation measures that could be pooled in groups, such as land use change, forest products, forest management, increasing productivity, and risk management. And the most important are measures with proven efficiency I will try to describe a little bit more. The most efficient measure ensuring the largest proven reduction of GHG emission is afforestation. But what is important to underline that afforestation is not always possible, and therefore continued emissions, especially methane emissions from flooded and rewetted areas, should be considered. And sometimes additional measures are needed to ensure climate change mitigation effect of afforestation. And these additional measures include proper soil preparation, use of appropriate or improved planting materials, fertilization with wood ash or complex fertilizers, wildfire prevention measures, and gradual felling or continuous cover forestry as a way of forest regeneration. Use of improved planting material is aimed to increase productivity and resilience of future forests. The effect can be estimated using forest growth models and standardized forest inventory data to obtain a local level activity data. And we found necessary to improve national land parcel information system to keep track of areas where measures are implemented. Although measures require some additional costs, at the moment there is no direct financial support for forest regeneration in Latvia. mounding soil scarification method increase CO2 removals and reduce loss due to natural disturbances, but long-term effect depends from further forest management steps. It is considered that short-term impact is relatively small. And the standwise forest inventory can provide information on areas where this measure is implemented, and thus long-term impact can be evaluated. And this measure is not supported at the moment financially in Latvia, but there is proposal to consider support for planting planting machines in Rural Development Program 2021-2026. Application of mineral fertilizers potentially is the most valuable measure in Latvia to reach 2030 climate targets. Linear additional increment rates are found according to the applied dosage of fertilizers, but estimation of long-term effects is not yet done. It's known that fertilization with mineral fertilizers increase N2O emissions from soil, but however, this effect is far less than net removals. And there is no support for fertilization in national legislation. However, it is not restricted either. And one of the measures that also results in additional increment in wood ash is wood ash recycling. And use of wood ash as fertilizer can be combined with mineral fertilizers to substitute phosphorus and potassium fertilizers. There is no models elaborated for Latvia to estimate impact of changes in forest management, and therefore only short-term impact is considered. Also, there is no support, financial support for wood ash recycling in forest land in national legislation at the moment. Fire prevention is a critical measure to retain carbon pools developed by other measures. Method for quantitative assessment are not developed yet, as well as cost-benefits ratio is not estimated yet in Latvia. But in additional prevention of forest fires, avoid emissions of harmful substances like PAHs and dioxins and heavy metals and other, thus contributing to maintenance of a healthy environment. And maintenance of fire prevention systems is supported in Latvia at national level by Rural Development Program. Gradual regenerative felling avoids increase of groundwater level and thus also methane emissions after felling. But in spite of potential benefits of this method, quantitative assessment is not done due to multiple constraints, for instance, root rot distribution in spruce stands. And what we can recommend is that there should be potential of strip harvesting in pine stands and setting area limits for clear fellings. The measure is not supported by legal acts at the moment, but can be applied voluntarily by forest owners. And in conclusion, and in additionally to previously mentioned, I have to point that for some climate change mitigation measures, controversial results are obtained, for example, on revetting, and it highlights knowledge gaps and missing activity data. National policy and climate change mitigation strategy recognize the role of organic soils in the reduction of GHG emissions, but however, no measures are directly addressed to organic soils. Mitigation measures in national policies lacks quantitative assessment as well as quantitative targets. And the most of identified measures are not soil specific. However, the effects may differ in mineral and organic soil. And as I already mentioned previously, National Land Parcel Information System soil and moisture maps should be improved to ensure ability to estimate and to project the impact of proposed climate change mitigation measures. And of course, country-specific methodology should be elaborated. So thank you for your attention. And this is it from my side.
Thank you, Arta. Any questions from listeners? Okay, then I will start. Can you go to one slide before conclusions?
This one?
Yes, about this gradual regenerative felling. Can you explain the principle? Basically, is it meant like that you cut some trees, take the biomass out? Well, basically partial logging, you take them out, but leave some of the trees in order to keep this groundwater level at constant point, so to say.
Exactly like this, yes.
Oh, okay, thank you.
In other words, it is a continuous forest cover felling.
Mm-hmm. But are there any like calculations how you can determine how much you can take out and how much you have to leave in order for this groundwater level not to raise or drop significantly?
I know that some studies are conducted in Finland, but in Latvia, no, at the moment we don't have any quantitative assessment of this impact of this measure. Impact on GHG emissions.
Okay, so it's at the start point.
Yes.
Yeah. Okay. Thank you. So, and anybody else wants to ask something, Torta? It was really interesting intro for this mitigation measures. I will ask the next speaker then, Aigars Strubergs. Aigars, hello.
Hello.
So, please, the floor is yours.
Hello, my name is Aigars Strubergs. I am from the Department of Forestry, Forest Utilization in Latvian University, Life Science and Technologies. The topic of my article, evolution of the attendance of refresher course for forestry machine operators, right co-authors Andris Lazdiņš and Leonard Sissons. The aim of research is to find out how forest machine operators working in Latvia use opportunities to increase their professional skills and professional development course to increase productivity. In today, mechanized logging harvester productivity is affected by several factors. Factor that cannot be changed, though has three species: diameter and breast height, type of filling, terrain, and others. And factors that can be changed are mostly related to the behavior of the operators, including psycho-emotional state, speed of reaction, speed of decision making, and others. One of variables the readiness of the operator of the logging machine. Periodic operator training play a very important role in increasing productivity. Training can improve the skills of operators to perform certain activities. The study examines the attitude of operators towards refresher training and its impact on productivity. A survey of forest machine operators was conducted to find out of option of forest machine operators about the need for training and the benefits of the training. The survey consists in three blocks. First, the general information about the operator clarify. Second, questions related to in-service training process, and questions related to benefits of the training and recommendation. To be able to perform the mathematical processing of the survey, the variance of the answers to the questions of the second and series blocks have the survey are completed according to the Likert scale in a 6-point system from strongly disagree to strongly agree. The survey was distributed electronically to operators in logging companies. In the last 4 years, in-service training was conducted for 315 forest machine operators at the Forest Machine Operator Center. 147 forest machine operators participated in the survey and filled in the survey. According to the survey, the average age of operators is 33 years. After education, 18% of the surveyed operators had primary education, 20% had secondary education, 55% had secondary vocational education, and 6% had higher education. 57 of the surveyed operators had obtained a diploma in a forest machine operator. The average total length of service of operators in logging machines is 7.6 years. Due to the fact that operators of two types of harvesting machines work in logging, the operators of harvesters and forwarders are separated. Initially, operators are sorted into separate groups. There are 3 groups according to the age of the operators, up to 30 years, 30 to 40 years, and more than 41 years. After working in the last logging machine for up to 5 years and more than 5 years, and education obtained accordingly. The average length of service of the surveyed forward operators is in this machine was 6.24 years. As shown in the table, 55% of forwarded operators have less than 5 years of experience. 40% of the surveyed forwarder operators receive secondary vocational education. Of the surveyed forwarded operators, 6 operators in the age group of the 30 years have obtained a forest machine operator qualification diploma. The others have obtained a driver's license for the relevant category of tractors and further improved their professional qualification by working in a logging company and attending a refresher course. The average length of service in this machine of the surveyed harvester operators was 4.7 years. 62% of harvester operators have less than 5 years of work experience, and 65% have completed secondary vocational education. In the age group up to 30 years, 24 operators have obtained the qualification of the professional forest machine operator. In the age group 30 and 40 years, 4 operators have a professional qualification as a forest machine operator. Other operators have improved their professional skills by working in a logging company and attending a refresher course. Again, the perceived altitude of the operators towards completing the surveys and the consequent small number of respondents, the possible responses to the allegation were combined from 6 to 3. As a result, the answers agree, do not agree, and disagree were used in the further processing of data. There was a significant difference between the respondents of harvesters and forwarded operators to the statement whether the training course were attended on their own initiative or sent by employer. 30% of freight forwarders answered that they had attended the freshers course on their own initiative. 30% partially agreed with the statement, while the remaining 40% did not agree with the statement that they had attended the fresher course on their own initiative. Internal survey forwarding operator indicated that they had attended the refresher course at the initiative of the employer. 33% of the harvester agreed with the statement that they attended the training or their own initiative. 57 partially agreed, but 10% did not. 65% of the surveyed harvester operators fully agreed with the statement that the training was provided at the initiative of the employer. 28 partially agreed, but 7 did not wish the statement. The apparent discrepancy in the data shown in the figure is due to the fact that 30% of operators have attended course twice or more a year. One of the preconditions for the success of individual training is whether knowledge and skills are assessed before at the training. According to survey data, the evolution of harvester forwarder operators on the list of knowledge and skills before training did not differ significantly. 6 forwarder and 12 harvester operators need that their knowledge and skills had been tested before the training. However, 90% of harvester operators and 86% of forwarder operators confirmed or partially confirmed that their knowledge and skills were assessed by instructors. As a result, the instructor had an idea of operator skills and working methods. In the training process, the instructors paid increased attention to the skills that needed to be developed. The survey of operators revealed that the instructor instructed the operator on more productive working methods.
Igor, sorry for interruption. The 10 minutes are over. I would ask you to could you like summarize and go to conclusions, please. Thank you.
Conclusions, yeah. Okay, I don't.
Well, finish the idea, yeah. And we are just like, yeah.
Conclusion: creating a feeling in the survey is electronic environment is convenient and does not require financial investments. However, there is a relatively low response rate. Operators under the age of 30 and work with work experience on the respective machine under the age of 5 attended the qualification course more actively. Less active training courses are attended by operators over the age of 41 and with work experience on the respective machine over the age of 5 years. The study found that 30% of forward operators and 33% of harvester operators attended the fresher course in their initiative. All forward operators and 65 harvester operators confirmed that they attended the refresher course in the initiative of employer. And of the surveyed forward operators and 66 of the harvester operators positively assessed the training process and confirmed that they had acquired no new knowledge and skills during the training. Thank you.
Yeah, thank you, Igor. Listeners, are there any questions, please? Okay, I wanted to ask about this initiative from that. I understand that these operator, the younger the operator is, the more he has the initiative to attend these courses. But how is it in general? Are these courses? I understand it's not like basic training, but it's so to say to have more knowledge and more know-how and forgot the word, to upgrade the skills, so to say. Is it like that, these courses?
I don't understand the question.
Are these courses like meant as upgrade of skill or like basic training? So you can work on this technique.
Toms, maybe please question in coffee break.
Oh, okay.
English is not my strongly.
Yeah. Okay. No problem. Yeah, I can do that. Yeah. Thank you all for interesting presentations, and we actually managed to finish almost in time. So yes, thank you. I would very much like to thanks for this opportunity to lead this session, and yeah, this was first time for me. So thank you all, and we have coffee break till 2:30, and yeah, this session is therefore over, and we'll see you at the next session. Thank you.
My name is Sabine Eglit. I'm a PhD student of veterinary medicine, and I have the honor to be the chairperson of veterinary medicine and the agriculture science section. So some technical details for the speakers. You have 10 minutes for presentation, and afterwards 5 minutes for answering the questions and discussions. Just a small reminder that first of all, you share your computer screen and afterwards the PowerPoint presentation. And first speaker, I would like to introduce you with the first speaker. First speaker will be Ilze Mattīse van Houtena with the research retrospective analysis of 101 canine lymphoma cases diagnosed in surgical biopsies in Latvia 2011 and 2020. So Ilze, the floor is yours. You can start.
Okay. Thank you for introductions. I will talk today about the retrospective analysis of canine lymphoma cases diagnosed in surgical biopsies. Lymphoma is a very important tumor in veterinary patients because it's quite common. It's malignant. It derives from clonally expanded lymphocytes, and it constitutes 7 to 24% of all tumors in dogs. However, it is not a uniform disease, and it's because it is clinically and morphologically diverse, it also can be a diagnostic challenge. The classification of lymphoma uses various schemes, and the most common ones and most commonly used are based on anatomical location and distribution, and based on cellular features and immunophenotype, which is according to WHO guidelines. But however, there's no currently data about characteristics of lymphoma encountered in canine population in Latvia, basically because most of the clinics have their own databases, but they don't have opportunity to combine and review them. However, the fact that my pathology service serves 80% of all veterinarians in Latvia. It gives opportunity to review all these cases and to make some conclusions. So objectives were to determine the prevalence of lymphoma among surgical biopsy cases in Latvia during a 10-year period, in veterinary pathology service based in Latvia to determine breed, age, and sex of dogs affected by lymphoma and to determine most common forms of lymphoma in terms of where they're located and also morphologic type and grade, and compare these data with those that are in literature. Before we jump into results, I wanted to briefly introduce the basics for two common classifications of lymphoma, and one is based on location, and the categories are multicentric lymphoma, which is most common and presents with one or several enlarged peripheral lymph nodes, and that's about 75% of cases that are seen in clinics. The less common types, but still important, are alimentary lymphoma affecting GI, mediastinal lymphoma affecting mediastinal lymph nodes and thymus, and extranodal lymphomas, which is sort of mixed bag, which contains all the other locations. Some people also separate out skin and mucocutaneous lymphoma, hepatosplenic lymphoma, CNS lymphoma, and so on. So there is no super uniformity in this classification. What's the usefulness of this system? It is that it allows correlation between clinical signs and distribution of lymphoma, and that is the sort of the most commonly used in the clinics on site. The other one is lymphoma according to WHO classification, which uses histopathology to characterize the cell size, cellular distribution, mitotic rate to derive at this lymphoma subtype. And it also requires immunophenotyping to determine whether the lymphoma consists of T or B cells. So immunohistochemistry needs to be done. According to this, there are more than 30 different types of lymphoma in dogs. However, 80% of lymphomas fall into one of five categories, and of these, the biggest one is, and those veterinarians who are listening maybe need to remember, is large cell diffuse B-cell lymphoma. And so you'll hear about that more. Usefulness of this system is it allows to distinguish between high-grade and low-grade. Low-grade lymphomas are also called indolent, and that allows more accurate prognosis and choice of treatment. So for materials and methods, pretty straightforward. It's a database search for lymphoma or lymphosarcoma, which is a synonym. We determined the total number of canine biopsies for each year and total of 10 years. We retrieved data for breed, sex, age, clinical history, organs examined, diagnosis and comments. Dogs were divided in three categories for the age, and then the detailed analysis of an anatomical location and distribution, and cellular and tissue morphology was done and recorded. So the first question, is there an increase in prevalence of lymphoma in biopsy submissions? So now you have to take into account that this is not what's seen in clinics. Only a portion of lymphoma cases make to histopathological analysis in pathology lab. So we're looking at a subset, but you can see from 2011 and 2020, the number of submissions increased, and along that also lymphoma cases increased. But when looking at the percentage of lymphomas, we can see high fluctuation between 1.1% to 4.4%, with average of 2.3% of lymphomas diagnosed each year. But there's no trend for increased prevalence. So we're staying steady. Second aspect, what about age, sex, and breed of Latvian dogs affected by lymphoma? So first, the age group, we can see that the most most of the cases fall in this category of 5 to 10 years old. That's about 68%. The median age affected was 8 years, with a range of 2 to 13 years. This corresponds really well with what's reported literature, where it's also median age is 8 years. For sex, more male dogs were affected than females, so 58%. Those that we didn't know sex or wasn't reported, we didn't include in analysis. So there's a male predominance. What's interesting, and we don't, since there's no control population included for this epidemiological analysis, then we don't know whether 42% of females is perhaps representative of population. But what's interesting to note is that there has been suggestion that intact females have lesser risk of lymphomas, and our data perhaps may suggest that, especially we know that the neutering of female dogs is not that common. It's becoming more common in Latvia, but in this study period, it may not have been that common. So for breed, again, the dogs that did not, we didn't have signalment information about breed, they were excluded from percentage analysis. Mixed breed dogs were most common group, but where we knew the breed, the three top breeds were Rottweiler, American Staffordshire Terrier, and French Bulldog. With all three breeds, that constitutes about 20% of cases. So so we could say that these are the top breeds in Latvia. In literature, we hear mentioning Labrador retrievers and golden retrievers as being predisposed. And in the same literature, Dachshunds are said to have a decreased risk. We did not see in our data support for that. But then again, have to take into account that this is a pathology lab submissions, not on site in clinic. And each country also has some different trends in the favorite breeds. So, but that's our Latvian data. Now, let's move on. The question about what are the most common anatomical forms of lymphoma for Latvian dogs? So the number one is multicentric, so corresponds well with what I told you before. So multicentric affects one or multiple lymph nodes. It may also affect spleen. There were 55% cases like that, which is less than 75% cases that I mentioned earlier, but it's probably because large numbers of multicentric lymphomas are easily diagnosed in the clinic without histopathology. And you can see here in this column that veterinarians that submitted tissues were already suspicious about lymphoma in 71% case for this multicentric lymphoma. The other categories, and I have few more than I had showed you before, and there's alimentary, skin and mucocutaneous tissue, splenic, extranodal and mediastinal lymphoma. These are less common, but they present as diagnostic challenge because only like 20 to 25% cases veterinarians suspect lymphoma. Splenic lymphoma I put in a different category because if it affects spleen, it can have a much more favorable prognosis than this multicentric lymphoma because it is indolent and low grade. The subtypes, cellular features of multicentric lymphoma in these 55 cases, and take-home message in this one is that most of the Latvian dog multicentric lymphomas fall in the category where this diffuse large B-cell lymphoma would be, which is a diverse group of lymphomas, but potentially high grade. And again, we can, based on morphology, we can fish out some indolent lymphomas with a better prognosis, and they fall into these lower categories. Immunohistochemistry is very seldom done, so I don't have much data on that, and it is a sort of lack of this or the weakness of this study.
So you are running out of time. Just
yeah.
Yeah.
Okay. So elementary lymphoma are mainly present, I think maybe, I don't know, 1 to 2 minutes, yeah? Because I still have, I'm sure there won't be questions.
Okay.
So elementary lymphoma mainly in the intestine, but not only. For those that are in the intestine, 15 cases, they're more commonly in the small intestine than in large, and more commonly they involve entire wall than just mucosa. In the skin, in skin and mucocutaneous tissues, most of the lymphomas are epitheliotropic, and you can see the lymphocytes infiltrating epidermis here. And less commonly, they're non-epitheliotropic. These are very big challenge, these epitheliotropic lymphomas, because they can mimic dermatophytosis, they can mimic autoimmune skin disease, they can mimic some inflammation. So histopathology is important. So in conclusions, lymphoma in dogs in Latvia is heterogeneous disease. There was no evidence of increased prevalence over a 10-year period. Most affected dogs are middle age with slight male predominance. Multicentric lymphoma is predominant anatomical type of lymphoma. And within multicentric lymphoma, 2/3 are intermediate to large cell lymphomas corresponding to diffuse large cell lymphomas. But even without the HC, histopathological examination can provide useful information to the clinicians. And we recommend that all large and intermediate cell lymphomas should be further characterized by EHC. And most importantly, diagnostic investigation of potential lymphoma cases is very important to practice evidence-based medicine and demystify lymphoma in dogs, especially because this disease can be treated with chemotherapy. And that's it. I hope you listen carefully and you have time for a couple of questions. Thank you.
Yeah, thank you, Ilse, and for interesting presentations. Or anyone has a questions? Somebody wants to ask something? Maybe I could ask, maybe I didn't understand right. If there is a tendency to these lymphomas to increase in latest years or not?
No, there's no tendency to increase.
Mm-hmm. Thank you. Maybe you still have questions, or okay, if there's no questions, then we would like to say thank you to Ilse. We will go to the next speaker. So the next speaker will be me. Once again, so my name is Sabi Neglit. I'm a PhD student of veterinary medicine. My supervisor is a doctor of veterinary medicine, Aija Ilgaža. And today I would like to tell you about the probiotic mixture X feeding effect on the growth and development of the broiler chicken digestive tract. So the issue of antibiotic resistance has become more pressing in the latest decades. So the main use of antibiotics in poultry are the treatment and prevention of diseases as well as the growth promoters. But since 2006, the European Union has banned the use of human antibiotics in animal as a growth promoters. So the substitutes for antibiotics are being sought. And the beneficial effects of probiotics, and antidiphers, phytogenic substrates have been studied very actively. And so about the probiotics, probiotics improve, improve the development and the growth of pathogens, inhibits the development of the growth of the pathogens like Salmonella and Campylobacteria, improve the intestinal microflora and the histomorphology of the intestines, as improve the digestibility and usability of the feed, stimulate the immune system, as improve the growth performance, live weight gain, feed conversion, and development of the digestive tract. So the aim of the research of our study was to evaluate the effect of the mixture X of lactobacteria on development of the broiler chicken digestive tract. So the study was organized in 3 trials from April 2021 to December 2021. In each trial, we had 216 broiler chickens, 1-day-old broiler chickens, cross-roast 308, males and females randomly divided into groups, the control group and the probiotic group. The birds were placed in similar pens on a deep litter system of wood shavings. The dietary treatment was basal diet for the control group and the basal diet plus the mixture X of lactobacteria, 4 grams per 10 kilos for the probiotic group. And first drinking water was provided as a lipidum, and the broilers were fed with pre-starter diet from day 0 to day 7, starter diet, grower diet, and the finisher diet from day 25 till the end of the study. Broilers were raised till day 35. All broilers were weighted in the day of the placing, and afterwards on the day 1, 7, 14, 21, 28, and 35, all birds of both groups. And yes, and at the same days of the weighting days, 10 birds from each group was randomly selected, and the gastrointestinal tract was excised, the proventriculus, gizzard, and then the intestines, and weighted also those with content. So the results. The initial body weight for the probiotic and the control group is considered to be very good for the future development and did not differ between the probiotic and the control group. That means that output data will not affect the further results. The initial body weight and the size of the chicken are very important factor in broiler production. There are studies that chickens with high initial body weight gain significantly more weight in future than those chickens with low initial body weight. And also for the other weighting days weight, we see the tendency for the control group to gain weight slightly faster than the probiotic group, but basically body weight did not differ between the probiotic and the control groups on all weighting days. Similar results were obtained by other researchers when the mixture of lactobacillus strains on a basal diet did not increase live body weight. The relative weight of the gastrointestinal tract remains relatively high until day 7 of age in the probiotic group and the control group, but it decreases with increasing body weight, and this could be explained that the birds are growing and the relative weight of the digestive tract decrease with increasing the organs, bone weight, and the muscle weight. The relative weight of the proventriculus is slightly higher in the probiotic group than in the control group till day 14, but later in the age the weight is equalized between the groups, and in the control group slightly milder than in the probiotic group at the end of the study. Yes, the same with the relative weight of the gizzard. At day 21st is slightly higher in the probiotic group than in the control group, but does not differ on other weighting days. The relative weight of intestines increases to day 7 in the both groups and then gradually decreases until the end of the study. It could be explained that during the first week, week, the bird grows rapidly, and the digestive tract also grows very fast, because well-developed digestive tract of the chicken ensures the successful further development and growth of the bird. But after the 7th day, the mass of the organs and the muscle mass of the bird increases, so the relative weight of the intestines decreases. And in general, the weight of the relative gastrointestinal tract parts did not differ between the probiotic and the control group. Overall, this study actually achieved very good results in both groups. This could be explained by the fact that in a parable condition where the animal is not exposed to the risk of diseases, stress, and such it was in our case of our study, and the addition of probiotics to the feed didn't give the expected results. And the various factors that could affect the results have been described and also in the literature, such as the age, sex of the animal, as well as the microclimate condition, feed consumption, daily regimen, etc., etc. So, in conclusions, I would like to say that exploring other studies, as I said, we can conclude that the study achieved very, very good body weight results in both groups. As I said, it could be explained by the favorable microclimate, daily regime, and so on. Various trends were observed in the study, but overall, the weight of the chickens and the relative weight of the gastrointestinal tract parts did not differ between the experimental and the control groups. In order to evaluate the effect of mixture of Lactobacillus X on the development of the digestive tract of broiler chickens, the study with histological samples should be continued. So thank you for your question. Thank you for your attention, and you are welcome with your questions. Did anyone have some questions? Maybe. If not, then we can go forward with the next speaker. So the next speaker will be Zaiga Jansone. The research is the application of ground-based high-throughput phenotyping platform in serial breeding and review. Yes, so Zaiga, the floor is yours. You can start.
Hello, my name is Zaiga Jansone, and I am the PhD student from Faculty of Agriculture at Latvia University of Life Science and Technologies. I have two supervisors, one Māra Blēdere, and the second supervisor is Gundaga Putniece. I will present your review article, and the topic of presentation is application of drone-based high-throughput phenotyping platforms in serial breeding. Agriculture faces three challenges: climate change, resource depletion, and population growth, which can have a negative impact on the quantity and cost of agricultural production. Cereals are the most widely grown crops in Europe, and it is important to focus on the development of cereal breeding programs and the introduction of new technologies, including high-throughput phenotyping methods. The aim of this paper was to analyze the recent scientific literature on the current use of ground-based sensors applied in high-throughput phenotyping platforms for the assessment and analysis of morphological and physiological traits in cereals and for the selection of high-yielding genotypes. Information about high-throughput phenotyping methods was used from 55 scientific articles. With high-throughput phenotyping methods, breeders can to perform different assessment, morphological and physiological of cereal plant. Furthermore, these assessments can be done on the organ, plant, canopy, and even population levels. To assess different traits, different ground-based high-throughput phenotyping platforms have been developed equipped with one or most commonly several combined spectral sensors. The most widely used phenotyping tools in the breeding programs of cereal varieties are LIDAR and different types of cameras. These are RGB, multispectral, and thermal cameras. In this picture, you can see the image taken with the RGB camera. For each phenotyping sensor has its positive aspects. For example, the main advantage of using RGB cameras compared to spectral sensors are their relative ease of use, low acquisition and maintenance costs. Multispectral and hyperspectral cameras, on the other hand, can provide higher image resolution, but their costs are much higher than for RGB cameras. Hyperspectral cameras can detect various stress symptoms in plants, including early draw stress symptoms that cannot be assessed visually. Here you can see different cereal morphological traits and these traits assessment with high-throughput phenotyping tools in breeding programs. For example, these morphological traits are early vigor, canopy ground color, above ground biomass, plant height, and the leaf area index. It is also important to evaluate physiological traits in cereal plants. These traits, just as morphological traits, can be assessed with high-throughput phenotyping tools. And these physiological traits are canopy temperature, leaf senescence, nitrogen use efficiency. The literature indicates that each type of sensors has its optimal application time depending on the stage of plant development, as the accuracy of the measurements results can be affected by light reflection from the soil. When working with high-throughput phenotyping methods, depending on the type of sensor used, it is important to follow certain methodological principles. These are setting of the experiments, taking the measurements, processing the data, interpreting the results. And in conclusions, sensors used in ground-based high-throughput phenotyping platforms, including visible spectral and hyperspectral cameras, can be used to assess and analyze morphological and physiological traits and select high-yielding genotypes in cereal breeding programs. And high-throughput phenotyping patterns enable the breeder to evaluate and identify genotypes of interest more quickly and accurately at different stages of plant development, including early stage and in larger field and laboratory trials than traditional breeding methods. When working with high-throughput phenotyping platforms, depending on the type of sensor used, it is important to follow certain methodological principles. And in cereal breeding programs, especially under field conditions, further research is needed to identify accurate biomarkers for the identification of nitrogen use efficiency and high-yielding genotypes by high-throughput phenotyping platforms. Thank you for your attention.
If nobody had a question, then thank you, Zaiga, for your presentation. And we can go forward to the next presenter. And the next speaker will be Lasma Rabantehane. Is it right? Yes. So in the presentation, I review morphological and gel parameters used for the evolution of potato nitrogen use efficiency. Yes. So the last month, the floor is yours and you can start your presentation.
Good afternoon, dear conference participants. My name is Lasma Rabantehane, and I will introduce you to our review article about morphological and yield parameters used for evaluation of potato nitrogen use efficiency. Potato is known to be the third most important crop in the world by production and the fifth by consumption. And historically, potato production in Latvia has been a really significant branch of agriculture. But in the last 20 years, potato cultivation has decreased. And as you can see in the figure A, from early 2000s, the area of potato growth has decreased by 33,000 hectares. And in figure B, you can see that production has taken downward turn by 370,000 tons. There are various reasons why is that right now, but one of the reasons is that potatoes need abundant nitrogen supply, which is often ensured with large amounts of fertilizers that result in contamination. And that result in European legislation, Nitrate Directive and Water Framework Directive established limits on the nitrogen input in crop production in Europe. So today, one of the goals is to optimize the high nutrient efficiency in crops. New potato varieties need to be improved with nitrogen use efficiency and to reduce the environmental impact of potato production without affecting yield and quality. So the aim of this review was to find various morphological and physiological traits of potato crops that can be used in potato breeding for nitrogen use efficiency or NUE evaluation in order to create new varieties. I will switch to results and discussion, nitrogen use efficiency in this particular article is considered as the potato's ability to use uptake nitrogen into the development of canopy cover protein and starch content as well as yield. Different authors in different articles have described NUE really variously, nitrogen use efficiency or NUE can be divided into two main processes in plants. One is nitrogen uptake efficiency or DISNUPE, which describes how well plants uptake nitrogen from the soil. And the second one is nitrogen utilization efficiency or NUTE, which describes how well this uptake in nitrogen is used to develop yield or other quality parameters. There are different nitrogen use efficiency or NUE definitions. Mostly it depends on the type of crop that is used in experiments. Nitrogen use efficiency can be described as yield per unit of available nitrogen in the soil, including the residual nitrogen present, as well as fertilizers. Then you can describe NUE as the dry matter per unit of nitrogen in the soil. You can also calculate nitrogen as fresh tuber yield per available nitrogen in the soil, and you can also calculate agronomic NUE, which is tuber's dry matter in kilograms per total nitrogen available in the soil as well in kilograms. Different authors have showed a lot of aspects that we need to take into account when we try to move forward with this breeding strategy towards nitrogen use efficient genotypes. And one of them is that potato varieties with improved NUE will perform well under high and low nitrogen availability. And that shows a little parallels with yield stability experiments. We also need to take into account that genotype is not only thing that depends on this nitrogen use sufficient characteristic. We also need to take into account maturity types. In potatoes with good nitrogen use efficiency, nitrogen utilization efficiency or this NUTE will increase as crops available nitrogen decreases, because then increasing amount of nitrogen has a negative effect on NUE and NUTE. And this is the reasons why in different experiments, NUE is better detected in cases with the smaller amounts of nitrogen in the soil. The first trait that is usually described in field experiments is canopy development. Canopy cover can be used in the vegetation season for the evaluation of NUE, especially this area under canopy cover progress curve that you can see in figure 1. This model is developed by Khan in 2012 and uses 3 basic equations to describe canopy expansion in phase 1, maintenance in phase 2, and senescence in phase 3. This model strongly correlates with NUE in different articles as well as with yield. And what is interesting, that nitrogen efficient genotypes suitable, for example, for organic farming must have early canopy development with high NUE and NUTE but low amount of nitrogen in tubers. And that means that in this phase 1 in figure 1 needs to be a really rapid growth in the start of the vegetation season and really stable growth in phase 2. But often when we speak about potatoes, we are speaking about yield and starch development. Yield is a result of genotype environmental interactions and can't be exactly used as one of the NUE characteristics, because only 48% of variation in yield can be determined by differences in nitrogen availability. But as I already said, yield stability is one of the main things that indicates how genotypes will interact in different environments and different nitrogen availabilities. Also, the starch content is mostly described in context with NUTE, or this nitrogen utilization efficiency. And nitrogen metabolism show a close correlation with NUTE, which indicates different mechanisms of how potatoes respond to nitrogen deficiency. We don't often speak about protein content when we talk about potatoes, but potato protein patatin takes about 40% in potato tubers, and it is one of the main storage proteins that is negatively affected by lower available nitrogen in the soil, and potentially will tell us how good this potato will be stored after the harvest. The main method besides field testing is in vitro method, or NUE evaluation under controlled conditions, because field testing of potatoes allows only 1 or 2 trials per year, depending on the climate zone. In Latvia, it will be only just one trial per year. In vitro culture systems allows investigating many plants under highly controlled conditions, excluding different pathogen effects in a short period of time. Also, it is a great opportunity to monitor wood growth in different end supplies. Different authors in various articles has described that nitrogen use efficient potato genotypes will have larger lateral root development than others. And also they have indicated that in vitro system can correlate with yield stability of cultivars, not only in pot trials, but also in field trials for the investigation of different NUE parameters. So for conclusion, it is necessary to optimize the management and use of high NUE potato varieties as an alternative to the existing large amount of fertilizer applications. NUE can be calculated with different methods or detected by various traits of the potato crop. A very good predictor of NUE in field trials can be OCCPC model and possibly yield, but not by itself, but more like in yield stability. The in vitro system allows for the investigation of root development, which can usually be impossible in soil experiments, and the results can correlate with field trials. Thank you very much, and I hope that you have some questions.
Thank you, Lasma. I hope that somebody have a questions. Does anybody have? Okay, maybe I will try to ask the question. I don't understand nothing about this. As I understand from your presentation for potato, then main nutrition, like main fertilization is with the nitrogen, as I understand. Yeah. The main, there is other fertilization possibilities or other, like we can replace this nitrogen.
Nitrogen is the main, but we can use different sources of nitrogen. Mostly we are using the synthesized nitrogen, but we can use like natural from manure or other nitrogen. But what is the main point is that potatoes need a really abundant nitrogen. It's a lot of nitrogen to make a good yield. But these limitations in legislations and nitrate directives, they restrict of using them because potatoes have like really shallow root system, and mostly it just makes groundwater contaminations. Because when we are using this fertilizer, it not always is available in available form for this plant to take up and use to make yield or other parameters in that season.
Thank you for your answer. It was, I understand much more about it. So, if anybody have questions for Lasma? If not, then we could say thank you, Lasma, for your interesting presentation. It was really nice to listen it. So thank you very much.
Thank you.
Yeah. So all the speakers are spoken, so we could maybe finish this section. So thank you for everyone, speakers for interesting presentation and listeners for attention. It was nice to be here with you, with all of you in this veterinary medicine and agricultural science section. So I wish everyone good luck in the future. Researchers, yes, and such a small technical detail for the next section. Now we have an extra coffee break, and the next section will education, information, and communication technologies will start at 3:45. It's like a 15 minutes delay, and the first speaker will be Birta Sloka. So see you in the next section. So thank you, everybody.
Good afternoon, dear colleagues. The Latvian time is 3:45, and this is time for our session. My name is Signe Baliņa. I'm professor at the University of Latvia, and it is my great pleasure to chair this last session of our conference. And we have some small changes in our timetable, in our schedule. As you know, the first presenters, they can't take part in our conference, and I really hope that they will be healthy as quick as possible. And another one small change is that, thanks Professor Birta Sloka, we changed the schedule that me and Dr. Liena Viļņuma will be the first presenters, and then we follow the schedule. About requirements and about this process, as we all know, There are 15 minutes for each presentation, 10 minutes presentation, and 5 minutes for the short question and answer sessions. And please, if you have any question, if you have questions to presenters, please raise your virtual hand, and I will give you the virtual floor. Lene, could you please share your screen with our presentation? My name is Signe Baliņa, and together with my colleague from University of Latvia, Liena Viļņuma, and also with research team from Datorzinību centers, we made the research user experience based design method application in electronic medical research data collection tool development. And it is really our pleasure to present our results. One of the main challenges in medical research is to find new methods and new tools for the prevention and treatment of diseases. An important part of every medical research data collection, is data collection and data analysis. Healthcare is facing many strict restrictions which govern data collection and use, and data are being collected by using various methods and from various sources. Medical research data is usually also highly sensitive, and all documentation concerning data capture, storage, and processing permissions must be approved before starting the data collection activities. And our analysis on the ongoing medical research projects at the Institute of Clinical and Preventive Medicine of the University of Latvia clearly shows that effort for the data collection and its costs vary from project to project, but is quite significant. And efficient use of information technologies in the data collection process will allow to reduce costs and also to improve data quality, and not only to reduce costs, but also to reduce time. Therefore, our research goal was to develop a cloud-based tool for automation of data collection. And one of the tasks was analysis of the role of user experience as a facilitator of higher IT tool effectiveness. And therefore, Lene, I would like to give you the floor.
Thank you. And here I will briefly introduce the evolution of the focus of human-computer interaction, because these two concepts are closely related. So if we look at the discipline of human-computer interaction, it deals with the design of interactive computing systems for human use. And several decades ago, the user was seen as a passive individual trying to efficiently use a computer. And the notion that interactive systems should be designed according to the needs, capabilities, and preferences of end users was not really taken seriously. Designers and developers assume that they themselves are prototypical users and design systems according to their own preferences. And the quality of interaction was determined by objective metrics such as task completion, time on task, number of errors, and similar. But it all started to change in the first half of 1980s with the proliferation of affordable personal computers and also growing numbers of untrained users. With it came the realization that it would be more beneficial for everybody to design interactive systems around user preferences, not the other way around, forcing users to learn and adapt to the system. So all these developments led to those so-called user-centered turn. And the attitudes and approaches slowly changed. The individual is no longer a simply user, but a human with all the rich context, character, and background that each and every one of us have. So then the designers and developers try to put the human in the center of the design process. And another difference is that interaction quality considers the whole interaction experience, not just task performance. This approach became so widespread and popular that it was even codified into an international standard. And the adoption of this approach means that following 6 principles, the most significant of which are that users are involved throughout the design and development, and the design is driven and refined by user-centered evaluation and addresses the whole user experience. We can see 4 basic steps of the process in this diagram, starting with the context specification. User requirements must be balanced with organizational and business requirements. Then design solutions are produced and evaluated by experts and representative users. And these last 2 steps can be repeated several times until the desired result is achieved. But what is this desired result? From users' perspective, it is the quality of interaction. And if earlier conceptions of quality focused on performance dimensions that are observable and measurable, the user-centered turn and increased leisure usage broadens this view considerably. User experience includes all the user's emotions, preferences, perceptions, physical, psychological responses that occur before, during, and after use of interactive system. And these personal interpretations can influence the future interaction with the system, and also they can be communicated to other users with the potential of influencing their subjective experiences. Over to you, Professor.
Yes, thank you, Lene. One of the tasks is also to define functional requirements of data collection automation tool. And we find out that there are 3 main several aspects: the development of the research protocol, the generation of data registration application, and data acquisition from external data sources. And after analysis of research data collection process and the evaluation of the most popular existing software, the functional requirements were defined and divided in those 3 categories. The first category is the data set identification and data logistics description requirements. Please, next slide. And those requirements are related to research protocol development phase. And in this phase, the research team members must discuss and agree upon the relevant issues and tasks to minimize mistakes and in the research protocol. And the following requirements relate to this category. Firstly, it is development of data sets in the research protocols, the reuse of existing experience. And also in this process of preparing data sets, it is really, really necessary to provide their visualization that allows to demonstrate the potential of data input form. And ensuring the possibility to generate a part of the research protocol documentation by describing the data sets required for the research project. This also is really important for the first draft of the research protocol. And when this research protocol is approved, it is also really necessary to ensure an operational transition to the data capturing phase. And at this point, a fully operational data registration application is needed with user interface and with usage of all the best practices, how to develop this. And the last point is an easy regeneration of data collection application. In case of changes to the research project, it is also very, very important. And the next phase is the data required, for a research project if they are available in external data source. Please, the next slide. Then an automated data acquisition process can reduce the workload when dealing with this data. And this is also really highly important to ensure that the data import into the data collection, to ensure the import of data into the data collection tool from a pre-prepared file, and also to ensure the response to the personalized data request to the external system in the generated data registration capture system. And all above mentioned requirements were implemented in the data collection automation tool, which is evaluated in our research projects. And now Lena, please, about evaluation.
As we recall, the human-centered process diagram a couple of minutes earlier, the iterative evaluation is an important part of it. And design solutions are produced through a logical progression from previous designs or ideas. And prototypes are necessary to support this life cycle. And changes in the design can be made rapidly in response to user feedback so that major problems can be identified and corrected before the proper system development phase begins. Based on the literature analysis for this project, it was decided to use two-step prototyping, starting with a low-fidelity original prototype and further refining it to design a medium-fidelity prototype. Usability review sessions were organized remotely, and the recommendations covered the general usability aspects from field types, adaptive design, keyboard navigation, and also touch-sensitive interfaces. They were incorporated into a medium fidelity prototype that included some major improvements, devoting more attention to medium-sized devices, including tablet computers, and also partial interactivity was added to the prototype in order to allow some limited interaction for evaluation purposes. And this prototype was also tested with the users, encouraging them to follow the steps of typical user journey. And here in the next slide, we can see two screenshots. On the left side is one of the first sketches, low fidelity prototype representing the functionality, meaning that users could achieve the specified goals. How convenient it would be is another story. So, and on the right side, we see an improved prototype. We can see that it is cleaner, it's less cluttered. Breadcrumb navigation has been added so that users can clearly see where they are in the system hierarchy. Also, confusing filter labels have been replaced. Create new button has been replaced with an icon, and users can edit any entry by clicking on a link instead of a button. And this is just one view, just for demonstration purposes. And the whole process, screen by screen, step by step, was made more seamless. It should also be noted that all suggestions and recommendations were carefully reviewed and analyzed against the most widely used usability, user experience heuristics and guidelines. And here we see them arranged by categories. For example, this minimalist design is meant not just for decorative purposes, but to make data entry simpler and more straightforward, to minimize the cognitive load. We also took into account that users can be distracted by other tasks or events under time pressure or some other circumstances. So the less elements there are on the screen, the less is the possibility of errors. And another example is status visibility. We made sure, for example, that it is always clear if an entry is being edited or saved, for example. Thank you. Over to you, Professor.
Thank you, Lena. And yeah, we are really proud that during our study, we developed the prototype of such a tool, and we also assessed how effective is this tool in medical trial data collection process. And the main conclusion is the last one, that we really take experiment with ongoing project, and we found out that significant economy of research team time and resources for development and refinement of the research protocol datasets can be achieved. Thank you for your attention. We have very, very short time for some questions, if you have any of them. Thank you, Lena. Thank you, colleagues. I do not see any raised hand, virtual hand.
Now I have hand. I have two questions. One is, thank you very much. First, thank you very much for excellent and very interesting presentation. and interesting, very interesting topic. But maybe can you explain a little bit more? What does it mean, data set with visualization, how it is, just to understand that? And also, as you mentioned in one of those graphs, one of the last graphs, yes, it was also good error messages. What does it mean, good error messages?
Yeah, thank you, Professor. Lene, maybe you can start.
I can start with the second question. Good error messages is just a good practice in human-computer interaction. So, for example, if you enter a data in a form, for example, your personal code, and it expects like you have 6 digits and then 5 digits. And if you make a mistake, error message doesn't say just simply it's wrong or some red border or something, but it explains that it should consist of 6 and 5 digits. Or for example, an email address should consist an @ sign. They are informative. So these people under great stress, they need a proper informative error message. So where is this mistake so that they can correct it easily? We rectified some of them as well. Honestly, at the moment, I don't recall the details, but it is also one of the principles of good human-centered design, so that we can guide the user in order to rectify their mistakes and move on as smoothly and as seamlessly as possible.
Thank you. And about the data visualization, there are 2 ways. The first one is that this is a tool when we just look how will be what kind of forms we will use and what will be how to easier collect the data, and therefore it is very important to see also all values and how to better code and how to better visualize this information in forms. This is by data visualization.
Thank you very much.
Thank you, Professor. Thank you. And now I would like to give our virtual floor to our colleagues from Latvia University of Life Science and Technologies, Marina Troškova and Irena Katane. Ladies, floor is yours.
Okay, so good afternoon. My name is Marina Troškova. I'm a year 3 student of PhD program of Latvia University of Life Sciences and Technologies, and today I'm going to present the findings of our theoretical study, which was organized together with my scientific supervisor, Associate Professor Irena Katane. So the topic of the research is ecological approach as a basis for a higher education environment. The pandemic highlighted the problem of providing learning environment, including that of higher education, that supports and encourages students to independently learn and retrain, direct them towards the job market and lifelong education, and help them achieve success in life. Such learning environment integrates learning and work, is fair and inclusive, and focuses on the students' individual learning and competencies. It also includes the latest technologies used to improve effectiveness and access. So the relevance of the ecological approach was pointed out by the contemporary trend in education, providing education for sustainable development, which is based on the diversity of education, including higher education. So the diversity of universities is important not only for ensuring sustainability of higher education, but also in achieving excellence in studies and research. The ecological approach to education helps predict changes in environmental context, promoting ecological thinking in individuals and society as a whole, fostering environmentally friendly attitudes and actions, preventing the consequences of reckless actions committed by people, and creating friendly environments, focusing the development of personalities. In achieving sustainability in education, it's important to ensure that it is open, accessible, and respects special features of cultural environment. According to the ecological approach, a critical objective of education is to create a balance between sustainability and continuously rising contradictory and competitive differences and the flow of information, which often results in imbalance, conflict, and stress. Green thinking and making the environment green is predominant global context for sustainability, linking people, the planet, and profits. Two additional contexts are minor and regional, resilience and alternative. All of this encourages changes in the organizational culture of higher education, adding sustainability values in strategic planning and in academic and management activities. A profound and critical representation of the world's views, contradictions, and tensions on a global and regional scale is necessary to find common paths towards sustainability. The ecological approach to education studies what happens in education using the dimensions of time and space, the development of individuals, society, and education environment systems interacting with a changing environment. In order to understand the ecological approach in higher education, it's essential to start with the definition of environment. The environment of an individual is defined by the interaction of natural and artificial circumstances in which the individual achieves personal development as a natural and social being. These parts are mutually related. A shared space that is directly or indirectly accessible by the individual is a component of natural environment. The context of the social environment is created by society and social relations that enable the individual to achieve personal fulfillment as a social being. The theoretical study highlighted the multi-contextual approach as a basis for the educational environment and research, which is one of the manifestations of the ecological approach. So in the recent years, some changes in higher education took place. The public now expects universities to provide professional training to students with a view to the future, ensuring long-term success and ability to live and work in an ever-changing job market. Higher education has undergone changes because of this, meaning that education becomes certain, accredited, and supportive. The main attention is focused on uniting and integrating the job market and the universities. A few universities have updated their strategies to improve access for working students and underrepresented social groups, at the same time ensuring all students can be successful. New accreditation forms have appeared, such as short-term, competence-based, and stackable. A critical role is assigned to creating, maintaining, and developing a university environment that promotes self-managed learning by students. Theoretical research carried out that revealed the different views pertaining to the context of the environment. Researchers often propose their own classifications of the dimensions, contexts, and components of the learning environment associated with the topics of their research and the specific features of the problem they researched. There is no uniformity in the use of terminology. In explaining the ecological approach, the terms dimension, context, component, element of the environment are used as synonyms. This indicates a lack of a generally accepted definition for a higher education environment and classification of the context of that environment. So the researcher Yasmin believes that the structure of the environment consists of a physical, psychological, and socio-cultural component, which describes the conditions and resources for the development of personality. The structural information model includes the living environment of the individual and relationships among the people. This supportive environment is also emphasized. Other researchers emphasize the idea of higher education environment, which includes professional development of students as well as their socialization as future specialists. Studies of trends in the development of universities show that the ecological approach is a priority in the activities of universities, as it activates the creative potential, competence, and competitiveness of students. So the researcher Heemstra defines the university study environment as an environment that consists of physical surroundings, psychological and emotional circumstances, and social cultural factors, all of which affect the development of a person or adult. Other researchers define it slightly different, believing that it consists of social, physical, psychosocial, and teaching components. The study environment where learning takes place, it affects the attitude and achievements of the students and makes this possible to organically feel the learning experience for students in higher education. Australian researchers highlight the physical, teaching, and technological components. In the context of the physical component, they mentioned the spaces that promote collaboration in teaching and learning processes, which include both internal and external features using video conferencing and accessing network spaces. The design of such spaces is considered to be such that provide the opportunities for teaching and learning, not provided by traditional learning spaces and classrooms. The advanced concept teaching space represents the state of the art in which the teaching and learning space, the use of information technologies, and the design of the study environment play the central role in teaching. Providing informal study spaces in a university has a positive effect on the students' learning process because these spaces enable students to learn independently or in groups, take breaks, or simply spend their time at university with their peers or communicate with their teachers. Fourth generation libraries also play a crucial role in creating the university environment because they currently provide not only different sources of different resources from printed to digital ones of information, but also provide study spaces for individuals and groups. In terms of physical environment, 4 design principles are highlighted: student focus, compatibility, flexibility, and accessibility. Generation Z prefers open spaces that promote active learning, as these spaces open up room for students and staff members to move about, creating a shared social space. The teaching environment component includes the activities, tools, resources, methods, strategies, and structures that facilitate the teaching of students. The psychological and social components are closely associated with each other, characterized by personalization, inclusion, student cohesion, satisfaction, task orientation, innovation, individualization, investigation, cooperation, student capital, and support by instructors. The learning environment and the individual learning process cannot be separated, as they continuously interact, complementing each other. And between the teaching and learning, there is a student's perception of the teaching and assessment processes. The perception of the learning environment affects the learning performance of the student. So some of the challenges have to do with the use of new teaching model, transitioning from the instruction paradigm to learning paradigm, with the rapid development of education technologies, with the diversity of non-traditional groups of students that need flexible courses, and with the rising hopes associated with the skills necessary in current and future professional life. New hopes associated with learning goals arise as well. Students are expected to learn the 21st century skills and skills for their current and future professional environment, in addition to the traditional content of education. So 21st century skills include creative and critical thinking, cooperation and communication skills, social and cultural competencies, problem-solving skills, and skills using information and communication technologies. In order to integrate the learning of these skills into curriculum, one must create an environment in which students actively learn using ICT and learn as experts. So another significant change in higher education involves the use of ICT. So it plays an important role, including such elements as blended learning, massive open online courses, and flipped classroom strategies with online video materials, which make higher education more flexible and broadly accessible. So the future, in accordance with the report, Universities Without Walls vision for 2030, the future universities will be hybrid in nature and structure. Universities will accommodate equally important physical and virtual environments at the same time. In the future, physical and digital learning environment and research environment will be united in a holistic way to meet diverse social needs and to enable the development of flexible and mixed approaches. The physical environment will continue to have a decisive role in social interaction and dialogue, a place for holding meetings that challenges and at the same time inspires, but also offers quiet spaces for focused learning and research. The virtual environment will make universities omnipresent. It will be created to improve every person's access to research and learning, to improve cooperation, to research new innovative methods, and pursue the missions of universities. In order to promote the professional growth of the student and their readiness for professional activities, the student must have an appropriate study environment. So the study environment of university consists of two key components: the environment of the university, and also the environment where the internship takes place. So the dual study environment of our university includes an interdisciplinary cooperation system and an interpersonal cooperation system. A student-centered study environment and student-centered activities improve learning, help students...
Sorry, Marina. Please go to conclusions part.
Okay, sorry. Okay, so to conclude, the ecological approach has been present in social sciences, including education sciences, since the 20th century, and is one of the critical components of creating and developing the learning environment. The ecological approach is relevant at all levels of education, including higher education, and according to the ecological approach, a critical objective of education is to create a balance between sustainability and continuously rising contradictory and competitive differences, the flow of information, which often results in imbalance, conflict, and stress. So the theoretical study revealed different views pertaining to the context of the environment. There is no uniformity in the use of terminology. So the learning environment and the individual learning process cannot be separated, and they continuously interact, complementing each other. Between teaching and learning, there is a student's perception. And this makes it possible to define the university study environment as a multi-contextual environment in which a special role is played to the ICT component. In order to promote a professional growth of the student and their preparedness for professional activity, the student must have an appropriate study environment. So a student-centered study environment and student-centered activities within it improve learning, help students gain specialized knowledge, and promote general development of skills and competencies. So the study environment creates possibilities for learning, and therefore the emphasis should rather be placed on creating a student-centered environment in which the student can use what is available. So thank you very much.
Thank you, Marina, for your presentation. I see that we have two questions, one from Professor Birta Sloka and the second from Professor Zelman. Yeah, Birta, please.
Yeah, sorry, I have several questions, just not one, but I have, so that what does it mean, new accreditation forms? What does it mean? Yes, because you just explained how it can be, what is related. Then what is blended learning? How, maybe in short, yes, and then no, okay. Third, maybe then later, if it is about this virtual environment and also physical environment, these connections. Yes, it was also somehow developed. But first, about these new accreditation forms, because no, this is.
Okay, so I guess what is meant by new accreditation forms, these are short-term, competence-based and stackable. So it means that, so previously people could just get the whole higher education or vocational education, but now they can make a kind of small steps. So first they can complete, let's say, first level of higher education, then they can take some courses, and then if they in a few years they can continue, and they can get a bachelor degree. So it's not like line based, but they can, let's say, build their learning, like it's made of small bricks, so it could be. Regarding...
but is it then the right word accreditation? Because accreditation, I think that it is for this institution, for study program, is it allowed or not allowed to provide this education? Yes, is it the same? So because it is not quite, because accreditation, it is not just, is it allowed and is it legally acceptable? No.
Okay, it could be a very good point, but maybe it's also meant that competence-based, it's also possible to recognize prior learning or prior working experience and get some credits for, let's say, higher education.
But then it is other word, because accreditation, it is just for that. Then maybe could be other word for instead of accreditation, because accreditation, it is just that it is that it is given permission to realize either study program or study programs, because then you can give this state recognized certificate diploma. Yes, but this maybe this word was very confusing. Thank you. Sorry. Mm-hmm. And okay. About this blended.
Mm-hmm. So by blended learning, I guess it has become quite topical in recent years. So it's like the combination of on-site and virtual studies, or on-site and remote studies. So blended learning. So that's what.
Is it so? Okay, that's very strange also. Thank you very much. About this, maybe about this terminology, this maybe it is. Because we know what does it mean, double-blind reviews for scientific papers that you do not know who is reviewer and reviewer does not know who is the author. But how it is about this blended learning? Yes, maybe how it is could be explained. If it is going to be, but what is going to be? How to understand what is that meant?
Thank you, Professor Slok. I think that there is a space to think about terminology and maybe to use some better terminology. Professor Zelma, please.
Yes, thank you. I would like to continue in some ways the previous question about the quality assurance, quality concept. How this concept of ecological environment is connected with concept of quality and quality evaluation, first of all, because quality evaluation is first step to receive accreditation, which you also discussed.
Mm-hmm. That's so. In our research, we haven't covered this topic, but I guess we can continue working on developing this and just include this question as well. So thank you.
Thank you, colleagues, for discussion, and thank you for presentation. Thank you, Marina. And now I would like to give floor to my colleague Birta Slovaka and to show the results of research about the role of education in reduction of income inequalities in Latvia. Birta, please.
About that a little bit what is also related to education and to very important aspects of education. It is that we know that just to reduce inequalities, yes, and in many cases it is just to reduce inequalities. Then there are the trade unions and also some other institutions, yes, who are just asking money from the government, just give. But we just here focused our research on that how to manage that also people, also those who are maybe a little bit more experienced in age groups, yes, who are older, that they can also be participants in the labor market, and and so that they can be involved also in especially that inequalities reduce by having better education. And for that, so that we were looking about different those aspects, yes, how it is used, especially this lifelong learning to increase employability for several groups at risk. It is young people, people before retirement age, and in retirement age. And this is especially we made that with analysis of different trends, and now it is more and more data it is available, and also we can have them, and that's about how to manage also this situation in our country, because it is really very sad that not all options are used, and not in so big extent as we would like to have. And here it is so that big challenges we have about this digitalization in the Baltic states, yes, which is recommended also by OECD. And so that also so we see that that share of population who are involved in this additional education, it is increasing, but not in so big extent as we would like to have. It is just approximately 4% or 5% of all these people who are involved in this additional education. And this is that one of these good things where it is possible to get additional skills and additional knowledge to increase employability, and then in this and as a result also decrease these inequalities in income inequalities. And here we have analyzed also how it is about different age groups, yes, that which are involved in additional education, and we see that mostly they are involved female persons, but in total it is increasing for almost all age group all age groups, and that it is just we can find these results, but still they are not different. There is big place where we can just improve, yes, and that is also that increases for men and female approximately the same, but men are less active in this extent. Even their payment is much higher, but so that the most active participants are those who are 25 to 35 years old. But we just there is a place of for where we can get better results, and especially it is necessary to pay attention to those who have this lower level education. So it is not those who have this level 3 and level 4 education, yes, and this is a little bit decreasing during the last years, so that it could be paid attention, because that is that during their formal education that they have got maybe not the most advanced tools for being competitive in labor force, yes, and there is also some place where it is possible to have that. Then also by gender, it's also that male are a little bit less active, and the female are more active also. So having, and also those who have less than primary education, basic or primary education, no, that's nice to see that it is also increasing that they are involved in additional these processes, and but no, in comparison with other countries, unfortunately, this share is quite low, and there is really big space to reduce inequalities, that people get to some education, yes, and that they can be more competitive. Now, about these households, because now it is in many extent also related to that, how people are equipped with internet tools and thus also other ICT tools. Now in our country it is really increasing, and no, so this our conclusions about this lifelong learning is a key for social inclusion. That we would like to stress by all our means. Yes, that it is really that just not to ask for additional support from the state, but just participate in these lifelong learning possibilities, because that is really just now offered in many cases, including also so-called work-based learning, which is developed by professional education institutions in good extent, and now they are also offering different additional education courses where could be also be involved much more extent in this lifelong learning. And this is in almost all those groups. It is possible, and it could be really realized, but especially for those who have this lower level education, that is with those who do not have this formal education. And about different how to involve those people who are just sometimes very passive and just looking only for material support from the government. Yes, it is really necessary to use different innovative social marketing and application of positive cases, publications, promotion, just to encourage people who are maybe not so well educated as they could be, but just also in all age groups, just start and do, because there is really good options right now, possibilities offered by the different institutions who provide this additional education, as well as there are really big share of population who really need that, that sometimes there is necessary to have these connections between those who are could be involved with those who are providing this very often really very good course, good possibilities. Thank you very much. It is not very short because we are already late. Thank you.
Thank you, Professor Slokas. Thank you. Please, colleagues, if you have any questions. I have one small question.
Yeah.
What we can see from your charts, that in last years there is increase of activities in lifelong learning. How do you think? What is the main reasons? Those online possibilities, or maybe state support for lifelong learning?
It was by state support. It was by rather big state support, and know that state support was provided by these European funds that it could be used, yes, for adult education and additional education, and that's, I think, really very good. And I think it's better to give a tool that people can be involved in labor market and can be productive, because if we look about this income, because now it is available, all salaries available which are paid by which are paid for the work, we can see that even for in age groups which are 70 and 74, even that they are also rather big share who earn really a lot, even 6,000 euros per month and more. And there is also no increasing also those who are maybe, because now we need to pay attention to this silver economy, that we need to involve in labor market also those who are maybe not so very young.
Thank you, Professor. Thank you. And now it's time for our last presentation. I would like to ask Natalia Sergeyeva, Yulia Mangal, or Kristaps Vergins to share your presentation about evaluation of meeting effectiveness for improvement of digital tools. Thank you. Floor is yours.
Thank you, Signe. Yes, as you see, there is a common work with my colleague Julia Mangale from Latvia University of Life Sciences and Technologies and Kristaps Vergins from company ViaDots. As we know, communication between company employers is one of the most important aspects in every organization. And data show that managers of large companies spend more than 75% of their time preparing, attending, and managing meetings. And since meeting is a place where information is exchanged, idea generated, and decisions are made, it is very important that meetings are organized as efficiently as possible. And that's why the aim of this study of our research is to analyze previous meeting dependence from other factors and meeting organizers' readiness pay for the digital meeting management tool. And now about our research. In the middle of 2020, company ViaDots developed the tool Meeting, a meeting management solution which helps to plan, manage, and follow on meetings and its results. In autumn of 2020, company ViaDots, with cooperation of our university, organized a study in order to find out efficiency, usability of this tool, and the possibility to improve functional requirements and economical usefulness. And of course, in order to achieve these goals, a customer identification survey was developed. A survey was conducted for a specific focus group selected by customer, and survey questions, of course, were divided into several groups. Description of company, previous company's experience in organizing meeting, and experience of respondents of using digital tools for meetings. The obtained data were analyzed using quantitative methods. The chi-square independent test was used to determine if these factors significant relationship because two categorical variables, and we are use principal component analysis to reduce the dimension of data set by transforming a larger set of variables into a small one. And now some our results and discussion. Respondents represent 5 sectors. As you can see, research and educational sector 27%, states administration sector 20%, and so on. Survey respondents most often represent company with which employ more than 50, yes, 50 employees, and the less represented companies, those which employ up to 10 employees. The analysis of the company's previous experience in organization meeting shows most of respondents indicated that they spent about 5 hours at meeting, and about 14% of respondents indicate that they spent about 15 hours at meetings during the week. Respondents from the non-governmental sector is the part who spent most of the time in meetings. Time spent in meetings in general correlates with the number of employees in the organization, and only 12.5 of the meetings always start on time, and there are no respondents who said that meetings always end at scheduled time. Analyzing the quality of previous meetings, respondents were asked to evaluate meetings from 1 to 10, and none one of respondents evaluated it with 10. Only 7% of all respondents evaluated with 9, but also none of respondents evaluate lower than with 5. And those working in public administration have given the highest rating, and those working in business and non-governmental sector have given the lowest rating. To evaluate survey respondents' previous meeting quality evaluation dependence from other factors, as I said, we use chi-square test of independence. And in our case, null hypothesis was a quality evaluation of previous meetings does not depend on the other factor, but alternative hypothesis, quality evaluation of previous meetings depends on the factor Y. And from this table, you can see that meeting quality significantly depends on the company's sector. Similarly, to evaluate survey respondents' readiness to pay for the meeting management tool, we also use chi-square test, and similar in this case, null hypothesis is respondents' readiness to pay for the meeting management tool does not depend on the other factors, but alternative hypothesis depends. And in this case, we get very interesting result that the readiness to pay for the meeting management tool depends on the end time of the meeting. The next step, as I said, we use principal component analysis method. And in this case, instead of the original 10 covariates, a data set of 4 principal components was estimated. In this case, PCY is company, PC2 meeting implementation, PC3 meeting results, and PC4 meeting planning. And in this table, we can see the component scores for different meeting evaluation. And a conclusion can be drawn that qualitative meeting depends of planning and simplified summarizing of the results. And in this table, we can see the component scores depending of the amount of the organizer would be ready to pay of the meeting management tool. And we can conclude that it depends of company sector and number of employees, and on ability to summarize result, number of meeting participation. And now our conclusions. The biggest points at the process of meeting organization is preparation of the meeting plan and preparation of the meeting minutes, as well as control of decisions taken and tasks set. Evaluation of the meeting quality depends significantly on the sector of the company, but readiness of meeting organizer to pay for the meeting management tool depends on the end of time of the meeting. In order to organize a qualitative meeting, planning of the meeting and summarizing of results is very essential, and readiness of companies to pay for the meeting management tool mostly depends on the sector of company, number of employees, the number of participants at the meeting, as well as the possibilities of summarizing the meeting's results. It's all. Thank you for your attention.
Thank you, Natalia, for your presentation and for your, I would like to say, very interesting and very impressive research. And yeah, colleagues, do you have any questions to Natalia and Julia? Okay. Yeah, Professor Sloka, please.
Yeah, maybe just how many respondents did you have? Because you told about shares in different fields, but how many respondents? Sorry that I have missed.
There was about 70 respondents from focus group selected by entrepreneur, because they asked these questions for potential clients.
But is it reason? Sorry, but then it's my next question. And is it reasonable then to apply this multivariate analysis, like as this principal component analysis, for so rather small amount of respondents? And is it then statistically significant, all those results? Thank you.
Thank you, Professor Sloka. I think this is. Thank you for your question. I think this is a good research questions, and maybe Natalia and Julia can think about this. And I would like to thank all participants for participation in this conference session. It was really my pleasure to chair this session. And yeah, I wish all of you success in future research and in future studies, and hopefully there will be also nice and hot spring also this year. Thank you for participation, and see all of you next time, hopefully on site, not only online. Thank you.
Yes.
Thank you very much. Thank you very much. Really wonderful. Thank you very much. Thank you very much.
Yes, I also would like to say that see you at the Latvia University of Life Science and Technologies in Jelgava Palace next year during the next Research for Rural Development conference. Thank you very much and have a nice evening.